Morning, everyone. We're going to go ahead and get started so we don't fall behind for this fabulous symposium. Before I go to the next slide, if you haven't yet scanned the QRS code or picked up one of the papers on the desk, that'll access you to the program guide for this morning's symposium. I'm Dr. Larry Eichenfield from the University of California, San Diego, and doing the honors today to introduce this symposium as president-elect of the IEC. So this should be a great session this morning with a phenomenal faculty who are world leaders to discuss decoding the dialogue, skin microbe, immune interactions, and atopic dermatitis. So the good thing is that this material for today is going to be recorded and available. The IEC symposium regularly are collected, so you should know that if there's material you'd like to review, or very importantly, if you have trainees or people who you work with, whether it be in science, clinical, translational research, know that there is a rich set of material that's been accumulated from the IEC symposiums. So this will be available on the website for viewing. If you have questions or comments, you can email them at info@eczemacouncil.org. And if you're interested in learning more about the International Eczema Council and what it does, you can send a note to Alan Stiles, who's our CEO. But I will describe now some aspect of where we're at with the International Eczema Council and how IEC does its things to try to positively influence the field of atopic dermatitis. So the IEC is an organization that's based upon the contributions of its core associates and counselors, and it's an international scope organization influencing atopic dermatitis. You can see we have 187 counselors and associates from 38 countries across the regions of the world. We're very conscious of trying to expand people who can influence atopic dermatitis from throughout the world and work on an international perspective. So we're very conscious of the global representation. You can see the 6 regions with North America, Asia Pacific, South America, Africa, Middle East, and Europe. We've been conscious to try to grow the leadership in atopic dermatitis in this way. So counselors essentially are established leaders in the field, either clinicians or researchers in atopic dermatitis management and research. There's criteria for judging someone's impact on atopic dermatitis to make it to the council level. And as a counselor, they participate in the IEC activities, not just the educational activities. There's a variety of work that's been done, position statements that have been done, research that has been done in partnership with other groups, and leadership in a broad set of scholarship activities. The associates are people who are younger than the more senior people, generally early career to mid-career individuals who clearly have shown their interest and work in atopic dermatitis. So it's a process where if you're interested, you can reach out to the IEC. We can tell you what's involved in the application process. You can work to figure out who you know who are IEC members already, because they can help to put sponsor levels. But the IEC continues to have its influence. Now, we have expanded the IEC membership to include both allergists, but also very importantly at the beginning of the SID meeting for PhD members as well. So we recognize the importance of individuals who are contributing to the research in atopic dermatitis with PhDs, and we've expanded the membership since last year to include these researchers. So the IEC mission is to improve patient care by promoting research education and best practices in treatment for atopic dermatitis in people worldwide. The vision is really transforming the lives of individuals and their families affected with atopic dermatitis. And our anthem, so to speak, is that we're globally focused. We're a global community of leaders in atopic dermatitis research, treatment, and prevention. And the IEC values are patient-centered, innovative, evidence-based, globally inclusive, collaborative, and looking forward. So a part of what we do is to host symposia such as this. It's been 10 years since the initiation of the IEC. There's 48 symposia that are presented. Again, many of them are available online. 155 unique expert speakers in 26 countries represented by the speakers. There's a repository of lots of information that's available through the web. We've had 146 videos, more than 100 hours of free content. So for instance, at the EADV last year, there was a superb discussion on cutaneous T-cell lymphoma and its relationship with atopic dermatitis. It was like the last session of the EADV, and so I know a lot of people had already left, but that's available online. It's a beautiful set of material, and certainly the symposia are a rich set of material, a state-of-the-art information. So this is our list of the symposia for 2026. We had a very successful joint symposium with the International Psoriasis Council and the IEC at the AAD. You see today we're doing our symposium now, and then next week the RADLA meeting in Buenos Aires. There'll be a discussion on AD in adults and diagnosis, misdiagnosis, and therapeutic approaches. September 9th, there is another joint session with the IPC in Heidelberg, Germany, as part of the ESDR. And then the EADV will usually do an annual symposium. The timing's being set up for that, and that'll look at the relevance of clinical trials and real-world data. We're proud of a recent accomplishment of the IEC, which relates to the publication with a group headed by Joe Marola that looked at coming up with a sort of standard definition for the verbiage to be used with low disease activity, minimal disease activity, on drug, control of disease, and a definition of remission. This took some work to do, but it's rather important because we've already seen clinical studies that have claimed remission with someone being off drug for 3 months and then going back on drug. I don't know if that makes it. So this panel did a lot of work to come up with standard definitions, and this article is an important one just recently published in JAMA Derm. Social media, please feel free to find and connect with the IEC. You can share our posts to amplify the good messages that IEC is putting out there about the importance of us continuing our work to advance the field of atopic dermatitis. We thank our supporters. Our corporate supporters have been very important in allowing us to put on symposia like this and doing the work that we do to try to positively influence the field. So our thanks to all of the supporters who are listed here. And without further ado, let's start the program. I'm very pleased to introduce April Armstrong and Pat Brunner from across the country ends, UCLA to Mount Sinai, who will be the program chairs.
Well, hello everyone. Thank you so much for being here. I know it's early in the morning, but Patrick and I, we promise that it's going to be a very high yield session full of learning and full of active discussion as well. We have really an action-packed agenda, and as Larry have said, that it's going to be really focused on skin microbiome and its interactions with AD, and I want you to really be prepared for all your questions for our speakers, and we really have also, you'll see from the agenda, really a wide variety of expertise that are represented here. So first of all, when you look at the agenda, and I'm going to go to the next slide, which has the program guide. So please take out your phones, and if you have a folder as well in front of you, but if you haven't already, you can access the program agenda with this QR code. So first, we're going to hear talks regarding barrier defect as a first step towards infection in AD. Then we're going to listen to how AD microbiome shapes the host defense. Then we're going to look at some of the innate immune defects. And then we're going to have a break where you get to also talk to our speakers further and network with one another. The key thing is do not leave this room. And then we're going to go to the second part of our symposium, which we're going to go over some of the cytokine programs that suppress microbial defense, and then looking at the immune endotypes and molecular risk profiles. And then finally, we're going to end these set talks with the infection risk across atopic dermatitis therapies. And then I'm going to have my wonderful partner, Dr. Berner, to introduce also the flash talks that we will have following that.
Well, thank you so much. It's a great pleasure to be here. Thanks so much for coming to our symposium. And yeah, without further ado, let's get started.
Great. Wonderful. And we can't wait to get started. And then for the flash talks, you're also going to hear about the epidermal, microbial, and spatial transcriptomic profiling, and as well as the psoriasis and atopic dermatitis transcriptomic profiling. There will be 2 topics on that too, so that will be very, very exciting. Okay, and now we're going to welcome onto the stage Dr. Teruaki Nakatsuji. And Dr. Nakatsuji is a microbiologist in the Department of Dermatology at UCSD, and his research focuses on understanding how the microbial ecosystem of human skin influences health and as well as disease, using a multidisciplinary approach that integrates biochemistry, molecular biology, cell biology, and animal models, and so forth. He investigates how dysregulation of skin microbiome contributes to barrier dysfunction, inflammation, and disease severity in atopic dermatitis. And his work has been successfully translated fundamental discoveries in skin microbiome research into microbiome-based therapeutics, now advancing in clinical trials. And with that, yes, I see, give him a big round of applause and welcome onto the stage.
Thank you, Dr. Armstrong, for the introduction, and thank you also this opportunity to give a talk. Today I will give the 3 main topics. So first, I'm going to talk about the host microbe interaction across the skin barrier, and the second topic I'm going to talk about the microbial mechanism of skin barrier disruption in atopic dermatitis. And the third topic is our most latest current work regarding a distinct microbial mechanism in the skin barrier disruption in the pediatric and the adult atopic dermatitis. So I'm going to talk about the first topic. So this is the gram staining of the normal human skin. So you can see a lot of the microorganism in on the surface, especially in the stratum corneum, and as well as in the appendage structure that's in the hair follicle. So this microbial ecosystem established the mutualism with the host. So that means that the both interaction, the host regulate the bacteria community, and the bacteria on the skin also actively influence the barrier integrity and the immunohomeostasis. So this is just a few examples of the function of the skin microbiome. So that they provide a direct antimicrobial barrier against the pathogenic bacteria, and also they help to maintain the barrier integrity by reducing the skin inflammation, and also the promoting wound healing. And some species of bacteria within the skin microbiome also influence the adaptive immune system. So that's why that in a balanced microbiome provide the more higher barrier integrity and the controlled immunity, and then the regulate the skin homeostasis. In contrast, in the imbalanced microbiome, we call the dysbiosis, that induce the barrier disruption and uncontrolled inflammation, and then they develop the skin disease such as atopic dermatitis. So the function of skin microbiome is mediated by a diverse cell types that reside underneath of the physical skin barrier made of the stratum corneum that's made of the dead keratinocyte. So previously, traditionally, so the skin commensal bacteria was completely separated from the host by the epidermal barrier. So if this assumption is true, so we have the one question: how the skin commensal bacteria influences such a diverse the cell types, the function of the diverse cell types. So actually we have a strong evidence indicating that the skin commensal bacteria existing not only the surface but also within the skin. So this is the immune staining of normal human skin with the gram-positive bacteria marker, anti-LTA IgG. So you can see the skin gram-positive bacteria exist not only on the stratum corneum, but also in the dermal area under the basal membrane of epidermis, like that. So when we conduct the in situ hybridization against the bacterial 16S RNA, ribosomal RNA, so we detected a lot of bacteria within the appendage structure and the eccrine gland. So this is not surprising, but the surprising finding is we detected the bacteria in the adipose tissue layer. So this our data suggesting that the bacteria routinely penetrate into the skin and then migrate into the position where the bacteria physically interact with the diverse cell types under the epidermal barrier. So next, our question is the atopic dermatitis is associated with abnormal bacterial penetration across the epidermal barrier because, as you know, atopic dermatitis is associated with the barrier disruption. So I don't need to explain this to this audience, but the culture-based and the DNA-based analysis indicate that this atopic dermatitis is associated with the dysbiosis that with the expanded Staphylococcus species, that the Staph epidermidis and the Staph aureus. So I will talk about this later, but these Staphylococcus species contribute to the barrier disruption in the atopic dermatitis. So this the image is the immunostaining of the non-regional and the regional skin of the patient with atopic dermatitis with the anti-Staph aureus IgG, so showing in the green color. So in the regional skin, you can see a lot of the Staph aureus colonizing within the epidermis and also in the dermis. But the stuff in the regional skin, Staph aureus signaling was not so much as well as not so much compared to the regional skin. So this data suggesting that Staph aureus directly penetrate into the skin and trigger the inflammation. So next we ask if what kind of barrier element control the bacteria penetration into the dermis. So because the atopic dermatitis is linked to the defect of innate immune barrier and also physical barrier defect, such as the filaggrin mutation. So that's why next we use the mouse model of the cathelicidin antimicrobial peptide knockout mice, and also filaggrin mutant mice that was sensitized, skin was sensitized with albumin to induce allergic inflammation. And we topically apply the Staph aureus onto the each skin conditions, and then obtain the epidermis and the dermis and adipose tissue by a microdissection, and then quantify the Staph aureus that enters into the epidermis and the dermis and adipose tissue by the qPCR. As a result, so the gene deletion of cathelicidin and also filaggrin mutations allows more Staph aureus get into the dermis and adipose tissue compared to the wild type skin. Okay, so so this result suggests that the cathelicidin antimicrobial peptide and the filaggrin is a kind of the barrier element that controls the bacteria entry. So next we examined if the barrier restoration can limit the Staph aureus entry into the skin. And so to address that, so that we use the same mouse model, filaggrin mutant mice, and then sensitized with albumin. So the allergic skin was allergic skin was treated with the barrier restoration formula of the lipid mixture. We call that epsilon. So that's composed of ceramide and cholesterol and fatty acid. And then we apply the Staph aureus. So as a result, epsilon significantly reduced entry of Staph aureus into the dermis and adipose tissue. So this suggests that the barrier restoration intervention may be the benefit to the patient with atopic dermatitis by limiting a Staph aureus entry. And also the epsilon intervention also suppressed the induction of IL-4, CXCL2, and IL-17A, the inflammatory cytokine induced by the Staph aureus application. And the second topic is the microbial mechanism of skin barrier disruption in atopic dermatitis. So to validate the observation of the Staph aureus entry into the mouse skin and also the human skin. So we just topically apply the Staph aureus onto the organotypic skin construct. So you can see a time-dependent pattern of the Staph aureus penetration into the deep in the skin. In contrast, when we apply the Staph aureus, the UV-killed Staph aureus on the skin, so the dead bacteria still retained on the surface, but not penetrate. So this data indicate that only a viable bacteria can penetrate into the skin. And this penetration is independent from the immunocell because this is the skin construct, no immunocells. So to address a mechanism of Staph aureus penetration into the skin, so we use the same filaggrin and oval mice model and then apply the three types of Staph aureus. So live wild type Staph aureus and the live mutant Staph aureus with the 10 extracellular protease mutant, the deficient. So this strain has no extracellular protease and also dead Staph aureus. So as a result, so the proteinase null mutant was not be able to penetrate into the dermis and adipose tissue like that, compared to the wild type Staph aureus. So this suggests that the importance of the Staph aureus protease in this mechanism. So a few years ago, the group of the Harvard University also made the very important finding. So they demonstrated that the V8 protease, that's the serine protease produced by Staph aureus, directly activate the sensory neuron in the skin through the cleavage of PAR1, that's the protease activated receptor 1. And this mechanism can induce the pruritus and also pruritus and also scratching behavior. So this study highlighted the Staph aureus protease not only the skin provide the Staph aureus protease so that the damage your skin through the direct proteolytic route and also the indirect route like that. So, however, the Staph aureus is not always on the atopic dermatitis skin. So this is the Staph abundance of the live Staph aureus in our AD cohort. So we found that almost 30% of the atopic dermatitis patients are the Staph aureus negative. So the next our question is what promote the barrier disruption in a Staph aureus negative atopic dermatitis. So we also focus on the other member of Staphylococcus species on the human skin, that's Staph epidermidis. So we found that the Staph epidermidis also increased in the AD regional skin. So, and we also found that the ECPA mRNA level on the AD regional skin is also elevated. So ECPA is a kind of a cysteine protease with a gelatinous activity produced by Staph epidermidis. So we found that the mRNA of this cysteine protease level on the skin significantly correlate with the local EG score on the patient. So we try to next validate this clinical trial in the mouse model. So when we apply the ECPA knockout mutant of Staph epidermidis on the mouse skin, so this mutant was not able to induce inflammation compared to the wild type the Staph epidermidis. And the mutant Staph epi couldn't induce the inflammatory cytokine, including IL-4, IL-6, and the 17A and the 22 compared to the wild type. So this illustration summarizes current our model. So the expansion of Staph epidermidis or Staph aureus turn on their chloram sensing, and then the activation of chloram sensing induce the protease and also other the microbial inflammatory mediators, including PSM and the delta toxin and the leukocidin. So the protease produced by Staphylococcus disrupt the skin barrier and induce abnormal skin entry over the Staphylococcus aureus or Staph epidermidis. And then induced the inflammatory mediator was detected by the sentinel cells, including a keratinocyte, a fibroblast, dendritic cell, and a mast cell, and also neurons. And then initiated Th17 and the Th2 mediated the inflammation. So, and then Dr. Gallo, who is giving the talk in this the session later, so he gonna give a talk what's happened in a transponder and the responder phase after barrier disruption. So the third topic is the distinct microbial mechanism in the skin barrier disruption in the pediatric and adult atopic dermatitis. So as you know, the pediatric atopic dermatitis is clinically different from adult disease. For example, the disease prevalence is much higher in the younger children compared to the adult. And also the pediatric atopic dermatitis involve more the facial skin So next, we just focus on the facial skin microbiome in the pediatric patient. So this is the shotgun metagenome sequencing from the facial skin swab obtained from the 2 to 36 months years, the 2 to 36 months old the infant or children with or without atopic dermatitis. So unlike adult atopic dermatitis, we couldn't detect a significant expansion of Staphylococcus species, at least in this age group of our cohort. So instead, we detected so diverse Streptococcus species. So I highlighted here. So these are the typical the oral associated Streptococcus species. So we also quantified the live Streptococcus on the skin surface by a culture-based method. So we found elevated Streptococcus survival on the AD regional skin compared to the non-regional and the non-AD control skin. So this difference was not detected by a shotgun genome sequencing. So, and please make sure that this data is shown on the log scale. So we also measure the protease activity of each Streptococcus isolate from the AD and the non-AD subject. We analyzed the total more than the 800 isolate from the 19 AD subject and the 16 normal subject. So we found that significant elevated the protease activity on the AD isolate compared to the non-AD isolate. So this data showed the protease activity from the Streptococcus salivarius isolate. So Streptococcus salivarius is the most dominant species found on the pediatric skin. And this data showed the big functional difference between the AD isolate and the non-AD isolate, even within the same bacteria species. So this data tells us the protease activity is a kind of the strain dependent, not species dependent. So finally, we examined the effect of the Streptococcus protease on in the inflammation using the MC903 induced type 2 inflammation model. So mouse back skin was pre-treated with MC903 to induce type 2 inflammation first. And then we topically applied the AD isolate of Streptococcus salivarius with the high protease activity, or a non-AD isolate with a low protease activity. As a result, when we applied the protease high Streptococcus salivarius on the normal skin pre-treated with the vehicle, so we found a strong induction of IL-17A and also cathelicidin antimicrobial peptides. So this is the typical immune response against the barrier disruption. But the protease low strain didn't induce these genes. So in contrast, when we apply the same protease high strain on the MC903 treated skin, so we found a strong induction of IL-4 expression, like that, rather than IL-17. And also the cathelicidin, the gene expression was partially suppressed. Importantly, this immune response against the protease high Streptococcus is very similar to the immunophenotype of the pediatric atopic dermatitis. So in summary, so we detected the different distinct dysbiosis pattern between the pediatric and the adult atopic dermatitis. But in the adult atopic dermatitis, Staphylococcus proteus played a dominant role on the barrier disruption. In contrast, so we found that the Streptococcus can mimic the function of the Staph aureus in the pediatric atopic dermatitis. So before closing my talk, so I want to emphasize, I want to give information about our relevant presentation in this meeting, SID meeting. So I'm going to talk about the function of gelatinous E produced by Enterococcus faecalis in the pathophysiology of pediatric atopic dermatitis. And the Carla Chaheda, she is the medical student in our lab. She's going to talk about the microbiome maturation from children with a high streptococcus to the adult with a low streptococcus. And also Yang Chen, she's a PhD student. She's gonna talk about the potential therapeutic strategy for the atopic dermatitis, targeting the expansion of streptococcus with a skin commensal species of Staphylococcus hominis isolate. And Tariq Wood, he's the MD PhD student. He's going to talk about the identification of Enterococcus faecalis in the metagenome sequencing data from the children's skin. And finally, I'd like to thank Dr. Rich Gallo, Dr. Tisa Hata, and Dr. George Hightower, who are the key collaborators of this project. Thank you very much.
Great, thank you very much. The talk is now open for questions. Go ahead.
Thank you very much for your lecture. You really learned a lot. I have two small questions for you.
Can you use a microphone?
I have two questions for you. For the first one, I noticed you mentioned the adipose tissue is decreased for the flag mutation mass. It's very interesting for me, as we know for the adipose tissue is more than the, for our temperature, they are much multifunctional. I'm just wondering, have you investigated whether the adipose tissue has other, the interaction with the immune cell or keratinocytes, other cells, except for the SRS in the AD pathogenesis?
Okay. Yeah.
Okay. For the second question, it's interesting you compare the dysbiosis between the pediatric and the adult AD. As we know, the elderly AD is also widely well known for us. I'm wondering, have you compared the dysbiosis with among the elderly patients with this pediatric and adult AD? Whether there's some difference among these three kinds of AD? Thanks.
Okay.
That's my two questions.
So regarding the first question,
Okay.
so we surprised when we detected the bacteria in the adipose tissue. So that's another project. So we found that so once the adipose tissue detects the Staph aureus, so that produces a very high amount of the cathelicidin and then contribute to the defense in against the bacteria invasion. So actually that's published in the Science in 2015, I think.
Okay.
Yes, and about the second question is
It's about the dysbiosis among pediatric AD, adult AD, and elderly AD.
Okay.
Have you investigated them?
So I didn't study the elder AD, but so we just can imagine how the oral microbiome influences influence the skin microbiome because of the flow through the saliva. So we detected a lot of the oral species on the skin, and we found that some protests from the streptococcus disrupted the skin barrier and then promoted the atopic dermatitis, I think.
Okay, I see.
Yeah.
Thanks.
Hi, very interesting work, thank you. A lot of this work is before treating with sort of type 2 inhibitors. Is it known what happens in either mouse models or humans? What happens to all of these bacteria after something like dupilumab treatment or any other type 2 treatments?
Okay, so actually in the adult skin, so the dupilumab significantly decreases the Staph aureus. We think the blocking type 2 inflammation restore the induction of a cathelicidin antimicrobial peptide and also other beta defensins, and then normalize the innate immune system, and then decrease the Staph aureus. But I also think the similar mechanism in the pediatric atopic dermatitis, but we still didn't confirm that.
Thank you.
Hi, this was a super cool talk. I'm wondering when you were looking at kind of like penetration of Staph aureus into the skin, I know you mentioned using qPCR and in situ hybridization. I'm curious if you think that the Staph aureus is living and proliferating and growing in the deeper layers of the skin, or if you think that it's entering and perhaps being killed by keratinocytes or immune cells.
So we confirmed that if the bacteria dead, so that we couldn't detect the bacteria by in situ hybridization because RNA is very fragile after bacteria dead. So that's why, so we can say that the bacteria detected by in situ hybridization may be the metabolically active. But I haven't confirmed if they alive or not.
Yeah, I think it would be very interesting to see if they're growing or proliferating in that area.
Thank you for the comment.
Thank you.
Tara, great talk and great summary. You know, I think we keep, in pediatric atopic dermatitis, we keep going earlier to try to figure out about what comes first. And certainly there's a lot of facial dermatitis that we see in atopic dermatitis, as well as a fair amount of facial dermatitis in children who don't get the atopic dermatitis label. But I guess one of the questions is, what do you think the sequence is in terms of, we say, we talk about the face as being a high traffic area because of drool, et cetera. One of the reasons there's a high prevalence of dermatitis in AD patients. But do you think it's a colonization effect? That they just, or there's open skin and at some point the strep species get going? Or do you think it's something different in that, in the question of which comes first, the core inflammation, which of course is a mixture of both environmental triggers and the immune system?
Yeah, that's the most difficult question to answer. Yeah, that's the chicken and egg question. Yeah. And I think at least the oral microbiome influences the barrier disruption stage. So I still don't know, but my assumption is that get the inflammation, and the inflammatory condition select the streptococcus community for the high protease activity. Only high protease activity strain can survive, and the protease from streptococcus might disrupt the skin barrier. That's my model I'm thinking.
I like that.
Yeah, thank you.
Great. Thank you very much.
Yeah, thank you very much.
Well, I love the questions, so keep them coming. This is the SID meeting, so for those of you, I do see some younger folks in the audience. I've always thought of SID meetings as traditionally lots of inquiry, lots of active discussion. So really continue to encourage that as our next speakers come up. Next, I would like to introduce Dr. Zeyang Shen, who is an assistant professor in the School of Molecular Biosciences at Washington State University in Pullman, Washington. And he is a computational biologist whose research lies in the intersection of microbiome science, genomics, bioinformatics, and machine learning. And Dr. Shen, you can come up onto the stage so people see who you are. And he has made significant contributions in terms of especially how microbiome contributes to human disease in AD. He's a recipient of the Washington State University's Presidential Big Ideas Initiative Award, which is highly competitive. And I think we are very fortunate to have a computational biologist here talking to us about his work in AD.
Thank you, Dr. Armstrong, for the very kind introduction. And it's my great honor to be here to talk about some of my work. Today, my talk is mostly about the work that I've been doing with my postdoc advisor, Dr. Julie Segre, since my postdoc. I just recently joined Washington State University last September. So, and like Dr. Armstrong mentioned, I've been trained as a computational biologist since my PhD. And I've just always wanted to apply my expertise in eczema research, also partly because I have a history of eczema myself. And now since that I'm running my own lab, I'm definitely wanted to do more eczema research, and hopefully you'll find something interesting from my talk and something that might be even useful for your research, and hopefully there could be real potential in collaborations in the future. So enough for the introduction. So let's get straight to the science. So today I'm going to talk about the skin microbiome and how they associate with AD. And also a slide for disclosure, so I don't have any conflicts of interest to disclose. So when we're talking about human microbiome, we're referring to these 1 billion to 1 trillion microbes living on the human skin. We all know that skin is our largest organ. It's also a habitat for a wide range of microbes, including bacteria, fungi, and viruses. And when we start studying the human microbiome, the immediate questions comes to mind is what microbes are there. So we're talking about this skin microbiome profiling. I think the first talk mentions about the shotgun metagenomic sequencing, so this is kind of a slightly bit more background in that. We have 2 mostly commonly used sequencing methods to profile the skin microbiome. One is the amplicon sequencing, which targets the conservative region, which is usually the 16S ribosome RNA for bacteria. But since we're only looking at these hundreds of base pairs and using these genetic variants in that hundreds of base pair to do taxonomic identification, we usually only are restricted to the genus level identification for amplicon sequencing. But the benefits of amplicon sequencing is that the cost is relatively low and the analysis is relatively simple. But on the other hand, we also have this shotgun metagenomic sequencing, which would sequence all the DNAs in a sample. So now we're not just talking about hundreds of base pairs, but millions of base pairs for the bacterial genomes. And because we're looking at all the genetic variations within these millions of base pairs, we can now get our taxonomic identification down to the species and even the strain level of resolution. But also the sequencing cost is significantly higher, computational analysis is more complicated, but since we're not just looking at the ribosome RNA, we look at all the genes across the whole genomes. Now we can study the functional genes and pathways. So shotgun sequencing can help us not just answer the question what microbes are there, but also what they might be doing. And as a computational biologist, I just love complicated data and more complicated analysis. So most of my projects and research are based on the shotgun sequencing data. Despite all these benefits of shotgun metagenomic sequencing, one challenge for analyzing the shotgun sequencing data is sometimes we lack the reference genomes to map the shotgun data, shotgun reads to. So if we mapped the human skin metagenomic data to the standard RefSeq genome database, we usually would end up with only 50% of the reads mapped, leaving the other 50% unmapped, which we sometimes call them the dark matter in the shotgun data. But thanks to the recent development of bioinformatics pipelines, we now have a robust method to assemble near-complete genomes from shotgun metagenomic reads. The overall idea is to first overlap the reads to get short context, and then we'll group the context based on their sequence coverage and genetic features into bins. And then we'll do several steps of the careful QC to get good quality metagenome assembled genomes, or MACs. And what people found is that when they integrate these MACs into the RefSeq database and do the mapping again, they would recover a lot of the dark matter and get the majority of the shotgun reads mapped. So this pipeline has been used in the human skin microbiome fields in several studies, and people have generated a few of these human skin microbial genome catalogs. But for all of these studies, they are mostly based on children's samples or adult samples, and none of them are based on the infant samples, which we know are, like the first talk mentioned, there's a higher prevalence for atopic dermatitis. So we started our research by creating this first infant skin microbial genome catalog. We call it Early Life Skin Genome Catalog, or ELSG. For this study, we basically applied the pipeline that I just mentioned to 565 infant skin metagenomes, and we ended up with over 9,000 prokaryotic MACs and over 200 fungal MACs. And when we compare these MACs with the existing microbial genomes in the database, we actually found 743 novel species that were previously not found on the human skin. So this large phylogenetics tree here summarizes all the 1,000 bacterial species included in the ELSG catalog. And for those with the inner branch colors that is not gray, indicate those 700 novel species. And with this ELSG catalog, we moved on to look at how the infant skin microbiome could be associated with atopic dermatitis. So to do this, we collaborated with a clinical team in Australia who did this vitality clinical trial. So during this trial, they first recruited thousands of babies when they were at 2 to 3 months, and they followed them through the first year of their life, and at 1 year time point, these babies were assessed for atopic diseases, including atopic dermatitis, food allergy, and food sensitization. We didn't get skin swabs from all thousands of babies, but for those 429 of those, we end up with 169 who were diagnosed with atopic dermatitis only at 1 year. 72 of them had both AD and food allergy. 30 of them had both AD and food sensitization, and 108 of them were not diagnosed with any of these diseases, so they were used as healthy controls in our downstream analysis. We also had access to some of the babies' skin swabs at 2 to 3 months when none of these were diagnosed with diseases yet. And from each one of these babies, we collected cheek samples and endocubital samples, and we analyzed these skin swabs through deep shotgun metagenomic sequencing. And for each metagenomic sequencing data, we mapped the reads to the ELSG catalog, and we end up with about 80% classification rate using ELSG, which is shown by those green boxes. And that's about 20% improvement over the standard RefSeq database shown by the blue boxes. Based on these highly classified reads, we started by asking what are the skin microbiome changes associated with atopic dermatitis. And what we found is that skin microbiome profiles can help us distinguish AD with and without co-occurring allergic diseases. So for example, we have Staphylococcus epidermidis here, the second one from the left, and we found So we all know that this species frequently associated with AD, but we only found this species has increased abundance on the affected skin of infants who had AD alone, and not significantly differential between healthy controls and those babies who had both AD and allergic diseases. On the other hand, the other taxon, the second one to the right, so since shotgun also not just captures bacteria but also fungi, we also have a fungal species, Malassezia sleuthiae, which is only enriched on the skin of babies who had both AD and food allergies. Another interesting taxon we found is this Lactococcus cremoris. This one is only depleted on the skin of infants who had both AD and food allergies. And turns out this is also the only taxon we found to have a negative correlation against the AD severity measured by the SCORAD score. So we believe this taxon could potentially have some protective role against atopic dermatitis. Then we asked the question whether the skin microbiome changes could be already happening even before the diseases occur. And the answer is yes. So when we look at those skin swaps from babies at 2 to 3 months, we look at their shotgun metagenomic sequencing data, and we saw these species already started to increase their abundance on the skin of infants who would later develop both AD and allergic diseases. So one of the example here is Staphylococcus aureus, which we know is another species frequently associated with AD, and we saw that this species abundance already has increased. Even before these babies can be diagnosed with atopic diseases later. But it's only for those babies who later develop both AD and food allergies, and not for those who would develop AD alone. So these data tell us that skin microbiome can not only help us stratify different AD outcomes depending on whether there's co-occurring allergic diseases, but might even serve as biomarkers that can help us predict the future AD outcomes. So far, all of these analyses are based on species level, and since the microbiome field is moving towards the strain level, we are also trying to answer the question whether there are strain level variations associated with atopic dermatitis. For those who are not familiar with the idea of strain, this is one of the analogies that I like. If I ask you what are shown in this photo, you might tell me that these are iPhones. But if I ask for more details, you might say that the iPhone on the left is iPhone 12, the iPhone on the right is an iPhone 15. And putting these ideas in the microbiome taxonomy, the brand would be equivalent to species, the models would be equivalent to strains. And just like different models could have different features and functions, different strains of the species could also have different genetic features that could alter the microbial functions. And to look at the strain level variations associated with AD, we developed a new bioinformatics pipeline that can help us identify the strain level variations associated with host phenotypes. So if you're familiar with the idea of GWAS, this is basically the same framework but applied to the microbiome data. So the idea of this pipeline is that we started with a bunch of shotgun metagenomic sequencing data. Some of them belong to group blue, some of them belong to group orange. And for each one of these data sets, we first map the shotgun reads to the reference microbial genome for species of interest and identify the genetic variations for each data set. And then for every genetic variation, we do statistical tests to compare them again between the two comparison groups. So in this whole example, the black variant is associated with the blue group. The white variant is associated with the orange group. The gray variant is not differential between the two comparison groups. So we apply this framework to our Vitality infant data, and we did see some strain level variations associated with atopic dermatitis. So for the example here on the top, this is a missense mutation in a fused gene of Staphylococcus aureus. And this mutation turns to have its reference allele associated with more severe AD cases, and the alternative allele associated with less severe cases or healthy controls. And then for the example here on the bottom, it's another missense mutation in PRS gene for Streptococcus thermophilus. It has the other way around where the reference allele is associated with less severe cases and alternative allele associated with more severe cases. So we're seeing these strain differences that could be associated with AD, and we're wondering what could be the source of these strains. And since we also have access to some of these babies' mothers' skin swabs, we process those samples using deep shotgun metagenome sequencing again and try to compare the strains we found on babies and those found on mothers. So here we used a strain tracking tool that can help us identify the strains based on the shotgun data using the NCBI genome collections. I applied this tool to several AD-associated species that I mentioned about, so Lactococcus and Staphylococcus. And here the x-axis represents the percentage of strains found on babies that are also found on mothers. And you see that the bars here, most of them are highest for 100%, which means that all of the strains found on the babies were also found on the mothers. But since we don't have like a lot of all of those mother samples, so there are some noises in the plots as well. And just using Staphylococcus aureus as a relatively simple example, because we often just found one Staph aureus strain on each individual. So here I ordered our samples based on their families and grouped them by family using the vertical gray lines separating the infant and mother samples. And then on the y-axis, these are the identified Staph aureus strains. And so the darker blue squares are indicates which strains are identified in which sample. And you see that usually we found the same Staph aureus strains in the same family for both babies and mothers, except for this family number 6, which where we seem to have a different strain for the babies and the mother. So overall, our data shows that there's significant strain sharing between babies and mothers within the same families, and we believe this could potentially contribute to some of the disease heritability for atopic dermatitis. So in conclusion, looking at this understudied population and skin environments can help us expand our view of skin microbiome diversity. And in the vitality study, we saw the skin microbiome profiles can help us stratify AD outcomes depending on whether there's co-occurring allergic diseases. And by doing this strain level analysis, we found these strain level variations that could potentially provide some mechanistic insights into how the skin microbiome could be contributing to atopic dermatitis. As a future direction, we would like to explore a bit more about this gut-skin axis in the infant atopic disease. We have access to the stool metabolomics data for the same infants in the vitality cohorts. And based on a very preliminary analysis, we saw strong signals in the gut microbiome associated with food allergy. And among those differential taxa, some of those are butyrate-producing bacteria, including this one, Roseburia intestinolis. And we know that butyrate can help alleviate food allergy, and also in our data, we saw that these butyrate-producing bacteria are more abundant in healthy compared to food allergies. So this is consistent with prior literature, but this study is still ongoing, and I'm looking forward to seeing what we'll find after integrating the gut and the skin microbiome data together. Another interesting thing going on in my own lab is that we are lucky to get this so-called WSU President's Big Ideas Initiatives to build a new microbiome center at WSU. So the whole idea is to integrate the resources scattered through on the WSU campus, including multiomics, bioinformatics, organoid, AI, commercialization, to hopefully have all the expertise on campus come together to help advance microbiomes research at Washington State University. And our goal is to use human health, especially eczema, as a testing field. And then hopefully the goal of this whole thing is to hopefully these resources can be useful for other microbiome labs on campus. Finally, I'd like to thank all of my lab members. I'm running a new lab, so I really appreciate these amazing people trusting me, and we're still actively recruiting. So if you or your students are interested in microbiome research or computation, especially computational biology, please let me know. And of course, I would like to thank my advisors and collaborators from the NIH and the Vitality team in Australia, and also my founding source from NIAMS and WSU. With that, thank you all very much for your attention, and I will be happy to take some questions.
Go ahead. Yeah, I see a question.
Hi. Thank you for your wonderful presentation. As we hear, all these samples are taken from families in Australia. Can we replicate these findings in other geographic locations? Like, in each geographic location, it has different plethora of microbiome. Is it applicable to be generalized all over, or we need to replicate it here in the United States or Middle East or in Europe? So can we replicate it?
That would require an independent cohort from the other geographic
So
locations.
maybe it's an opening for a new, in your new position in Washington, maybe a new replication of it for here in the United States.
I would love so.
Yeah.
That Vitality study, I think they took 10 years to follow these kids and collected all the samples. But if I have the luck and the funds to do that, I would love to do that.
Thank you. Thank you.
Great talk. Quick question. So when you have different ages at times, and the question is, are you able to evaluate if patients have been treated for their atopic dermatitis? For instance, looking at the malassezia species in the children with both food allergy and atopic dermatitis, I wonder about differential treatment, which may be influencing colonization, or is this, because this is like real life rather than a population that's not treated over time?
So first of all, when they were first recruited, like at early point, like 2 to 3 months, none of these babies have symptoms.
Yeah.
So,
Yeah.
But at 1 year, we cannot exclude like if some of these babies started the treatment. But for this cohort, this is real life, so it's kind of reflects the general population. So a lot of these kids were having sort of mild to moderate symptoms, so it's not really like severe enough to go through like very systematic treatments.
But just like when you showed the SCORAD and the impact, you do wonder whether the patients who had higher SCORAD are treated in a differential feature, either with antimicrobials or anti-inflammatories that may mediate what would be the natural colonization at that point. So it'd be interesting to see. We have no data on that. Yeah.
Treatment can definitely be a thing, but we don't have like all the data in terms of like what kind of treatments they have. So that's kind of the thing that we are not able to include in the analysis.
Hello, nice talk. I'm Luis Santamaria from Barcelona. I'm just thinking on the translational relevance. We are talking about microbiome. We have the AD, we know their work anti-IL-23, anti-IL-22, 13, 31. And what about the microbiota? Any plans to translate those data to real microbes and the effect of those on the key pathological mechanism of disease to confirm this results?
I think that's a great question, and I think that could be a better question for my clinical collaborators. As a computational biologist, I feel my job is sort of more present these hypotheses that I analyze from the data, but in terms of how they might be translated and can have like real impacts on like treatments or like preventing the diseases, I don't think I have the expertise to answer all of that. So.
Thank you very much for your great talk. I'm Jung Shin from Korea. My question is that when the family share the same strains, will they have the family history of the atopic dermatitis or other allergic disease? This is my first question. And the other one is that what's the determinant of the different strains? Is that environment or genetic? Or if we have one strain when we were young, will we have that strain for all the other life?
I think I missed the first question, but the second question, definitely. So there could be, I mean, mothers are keep holding the baby, so they're definitely, and they're living in the same household, so there are definitely environmental things that could be affecting that. Genetics, it could be, but since we're not genotyping all of these individuals, so it's hard for us to really say about those factors.
So like when the kids leave their home, will they have the different strain?
That could be a very interesting question to study, but it's outside the scope of this research, though.
Yeah, actually. And my first question was that when they share the same strain in the same family, will they have the kind of same allergic disease in the family?
So we did collected the mother's atopy history. I didn't see like statistically significance in terms of whether the strain sharing would be relevant to how the mothers and babies would have the same allergy outcomes, though. But that could also be because we don't have like tons of the mother's data in our cohort, so it could be something interesting to explore more. So.
Thank you.
Hi, very fascinating. I was just curious for the different allergic diseases and their correlation with AD, if you were able to see if there's specific subtypes of allergies that are related to the severity and like the different strains.
I think that would be a very interesting question I'd like to explore. For most of these babies, they have either peanut allergy or milk allergy, so it's not like a very diverse range of allergens for these babies that I have statistical power to test each one of those. So we kind of group them all together as food allergy in general.
Thank you.
Great. Thank you very much. And thank you, Dr. Shen, for that wonderful talk. By the way, you heard Dr. Shen is recruiting, so I would love to sit behind a computer and just do that all day. It's the patient part, pipetting part that's not so exciting to our trainees these days, but wonderful. And I am going to introduce the next speaker, whom I'm sure you have never heard of. So it's my pleasure to introduce Dr. Richard Gallo, who is Irma Gigli Endowed Chair and Distinguished Professor and the Founding Chair for the UCSD Department of Dermatology. He's also the president of SID. He's here because this is undoubtedly the most exciting session, and we are so happy to have him to share with us his expertise. His work, as we all know, have really revolutionized our knowledge of function of the skin microbiome, not only in atopic dermatitis that we're talking about, but also rosacea and acne. So, Dr. Gallo. The floor is yours.
Thank you, Dr. Armstrong, and the organizers for inviting me to speak to you all today. And thank you for the suggested title, which is so easy to cover in a few minutes. But I really liked it because it sort of triggered me to give more of an innate immune overview, history kind of talk. Because from my perspective, this is the origin of the microbiome field and understanding the potential of microbiome research in therapeutics in particular, atopic dermatitis, which I've always considered sort of the low-hanging fruit for microbiome therapy. So let's see, there we go. So I'm going to organize this in several short parts just to kind of put perspective in things. So first, what do we mean by innate immune defense? What are some of the fundamentals of this? And what innate immunity means is non-learned immunity. This is genetically encoded host defense processes. And understanding why all organisms need that is based in understanding how potential virulent pathogens operate. And when you think about a microbe attacking a mammal, microbes such as Strep pyogenes, group A, otherwise known as group A strep, or the flesh-eating bacteria, will divide in a permissive environment about every 20 minutes. And what that means is that if you're inoculated with as few as 15 strep, within just a few hours, you will be overwhelmed and dead if there was not some sort of innate process in that environment to regulate its division and expansion. And understanding what these innate antimicrobials are, or AMPs, in the modern era can be traced back to Alexander Fleming, that perhaps some of you recognize the name because a few years after his AMP discovery, he discovered penicillin. This is, kind of like stepping back thousands of years. A lot of natural herbal medicines that have developed, as far back recorded history as the Egyptians, were based on the fact that different animal and plant extracts would be potentially useful in wounds and to limit infections. And what Fleming found in 22 is that when he sneezed on a plate of bacteria, everywhere his nasal droppings hit the plate, it caused a lysis, or it killed those bacteria. So he named this activity lysozyme and went on to identify the protein, i.e., a few decades later, then know the gene for the first antimicrobial, which came from human translational medicine. Hans Bowman in the 80s was kind of the father of antimicrobials, and using biochemical techniques to try to purify what the protein products and then the genes were, he realized that using insects, which are obviously evolutionarily more ancient, would be a useful source, and he picked these giant moths about the size of my hand known as the Cecropia moth, ground them up and purified something known as cecropin. And then Bob Lara at UCLA wanted to understand more about how neutrophils killed microbes. Now we're getting a modern immunology era, and he was and his group were the first to discover the defensins and the many different types of defensins. Mike Zasloff, around that same period, noticed that frogs in terrariums that he was doing experiments on. And Mike is an MD pediatrician at University of Pennsylvania. So what he was doing with frogs in terrariums, I have no idea. But he was interested in wound repair and noticed that in those dirty tanks, the frogs never got infected, and then purified something known as meganin. And Zasloff and Bowman and Lehrer were kind of a gang of friends and enemies. One of the things Bowman did was to take different frogs and put them in different tanks and show that whichever tank had a particular type of frog, the frog would change the overall microbiome of the tank. Again, showing that there's these antimicrobial factors that are controlling the microbial environment. Then I got very lucky as a postdoc working with Mert Bernfield he said, hey, I just finished my dermatology residency, and he said, hey, you're a dermatologist, you're coming to a developmental biology lab, why don't you look at stuff in the skin that might be of interest in sort of barrier defense and repair? And we actually, somewhat by accident, discovered the cathelicidins as the first antimicrobials in mammals, and then went on, because we were lucky enough to do this in the genetic era, to be able to clone the genes and do the rest of that. And around the same time, Jules Hoffman and his team were looking at Drosophila, a very powerful genetic tool. And he found josomycin and a number of other pathways, and he won the Nobel Prize for this. So it was a very exciting kind of period that I was lucky enough to be kind of born in, and started our interest in the antimicrobials. Being able to clone the gene in mice, similar to what Jules did in Drosophila, but he could do it in like 6 months, whereas, for us, it took us 7 years. But we eventually were able to knock out the gene in mice and show, at the time, which was somewhat surprising to the rest of these guys working in frogs and flies, that something as evolutionarily ancient as these genes would still be so important in a advanced mammal with all of its adaptive immune responses, as well as its reactive oxygen and other innate immune responses from cells like neutrophils. And then also a very important finding in terms of understanding the antimicrobials on human skin came pretty recently from Dr. Nakasuchi, who spoke to you earlier this morning, working in our lab. And he was interested in understanding, are those microbes that live on the surface of the skin normally? And most of you refer to these microbes as commensals, which means one side is benefiting. The microbe in most cases can live there, so benefits from us. But it says nothing about the microbe benefiting us. And he went and through functional screening at high throughput was able to identify that many of the microbes that we think about as commensals were in fact mutualistic, and that they benefited us. And these are the antimicrobial ConS coagulase negative staph species, and he went on to identify the genes present in these microbes. So putting it all together, the field is now recognizes that at all barrier surfaces, there is a complex system of innate immunity that is dominated by the production of antimicrobial peptides that are special for different barrier surfaces. There's some overlap, but also some very distinct ones that exist in the colon compared to the skin. The cells have a very strict pattern of regulation, which is very important, and I'll explain why. And that in many cases, this is in cooperation with a mutualistic population of microbes that might live in the gut, might live in the skin, that microbiome that produces, in some cases, the antimicrobials. So this is one important point when talking about strains. What do we mean about a strain of a bacteria species? What we're talking about is one sort of variant within that species that has a very unique and specific genetic makeup. But for us, what matters about strain is not that it is unique, but what matters is what are the genes in that particular strain, and are they functional in either a beneficial or a negative way? So for us, up in this upper right-hand corner here, you see these commensal-derived amps. What we care about is the strain, because it contains the gene that we have gone through and others have gone through to identify could potentially be beneficial. And that'll be more obvious as I move along. So that's part one. Part two, I think it's helpful to understand a little bit about the mechanism of action, why they're doing what they're doing. And the important aspect of most of the skin antimicrobials is to understand that, one, yeah, they're an antibiotic, they'll kill, but two, they're kind of a very potent cytokine, and they have important immune responses, so that amplifies why they're still relevant in organisms such as mammals. The way they work in most cases as antimicrobials is very straightforward. They integrate with a microbial membrane. It could be a virus, it could be a fungus, it could be a bacteria, and penetrate that membrane so the cell can no longer divide or actually completely blows up. So its specificity is based on its structure and the lipid composition of the microbial membrane. The other important thing is that some of the cytokines, some of the amps act as cytokines. And in the case of LL37, which is also membrane active and disrupts microbial membranes, when it sees a mammalian membrane, depending on the cell type, it will activate cell receptors, not by binding directly to the receptor itself, but by changing the lipid raft environment around the receptor. So this is a receptor that is off, and when LL37 alters the membrane, this receptor turns on. And that's particularly true for a family of G-coupled protein receptors that are very potent signaling molecules. And one example of how that has helped us understand disease is in psoriasis. This is some work that was done by Lun Zhang in my lab several years ago, where she observed that the LL37 and beta interferon in the skin exactly co-localized in psoriatic skin. We knew LL37 was high, but we didn't understand what it was doing in the skin at that time. And what she saw is the possibility that LL37 may be influencing interferon production by the skin, and went on to experimentally demonstrate that. And what she showed is that when human keratinocytes see higher levels of LL37, they start producing interferon beta in response to natural occurring double-stranded RNAs. These are U1 RNAs. This is what the structure looks like. So this turns on actually an antiviral response, but also, unfortunately for psoriasis patients, those beta interferons, type 1 interferons, activate plasmacytoid dendritic cells, which then educate and amplify the Th17 immune response. Why this happens is kind of structurally interesting to me, maybe not everyone, but if you do X-ray crystallography, actually this is Sachs structure with a cyclotron, you see that the LL37 assembles in these packets, they grab DNAs and RNAs, and then they present it to the cell surface. So this is proving that this is a nucleus. It's not projecting pretty well, but there's a cell here, and we're looking if the U1 RNA is binding to the cell surface, and it's not. But if you put LL37 and the RNA there, the U1 then will bind to the cell surface. It's something called a proximity ligation assay. And this is some mutant control peptide showing that if it's the wrong structure, it won't bind. So the LL37 makes the structure and then allows the cells to see this. So to summarize the whole story, what you get is that antimicrobials like LL37 will defend the skin by and shape the microbiome by both direct and indirect methods. They can either directly kill bacteria through the antimicrobial response, or the LL37 can, through a couple of different mechanisms, activate interferons, activate the G-coupled protein receptors, and make an inflammatory response that leads to inflammation. And then the innate killer cells like neutrophils and so forth come in and start shaping the microbial community in a situation where there's a barrier rupture. Okay, so part 3, I think now getting more exciting is, so what does all of this basic immunology science have to do with us as physicians, and more importantly, how can it be used to influence our patients? And the bottom line here is that things like LL37, these types of amps, are bad and can cause disease in 2 ways. And this is just a limited list, but it's the ones we've worked on the most. In one case, if there's too high antimicrobials, such as we see in rosacea and psoriasis, that precipitates and amplifies immune responses. But in the case of atopic dermatitis, this is a situation where Michael Howell, who'll speak later, observed, and we observed, that the amps are too low. So that observation was first made in work we did in collaboration with Donald Leong, now quite a long time ago, published in New England Journal of Medicine, where we recognized the susceptibilities of certain types of pathogens, such as herpes virus and Staph aureus in patients with atopic dermatitis, and we found that somewhat logically these antimicrobials, which we believed were very important for antibacterial and viral defense, were in fact lower. This is one of the images from that paper showing that the human beta defensin 2 was much lower in atopic dermatitis inflamed skin than in psoriasis. And later work done in Dr. Tisahada's group also showed that atopic dermatitis compared to normal patients who have a wound was very low. So there was something not wrong with the antimicrobials themselves, but that they were not able to be properly induced when there was inflammation. They just came up a little bit, not enough to kill the Staph aureus. And then Dr. Nakasuchi has also gone on to show that not only are the mammalian genes not present, but the bacterial genes, those bacterial strains that are help fighting against Staph aureus, are also rare on atopic dermatitis patients. So we have a double defect here. We have a Th2-driven suppression of the expression of the host antimicrobials, and we have a unique Th2 environment situation where it suppresses the survival of the strains of coagulase-negative staph that are producing the genes we care about to cause defense. One other, this is work that's continuing going on, and the associations between disease and these molecules are really exciting. This is some recent work that was just published from Marcus Chan in my lab last year, where he found that psychological stress suppresses the ability of your skin to make AMPs. This goes by a somewhat different mechanism, and it was exciting. It was never known that TGF-beta was was a molecule that was stress-induced through the HPA axis, and TGF-beta acts to suppress the ability of cells in the dermis to produce antimicrobials. And this leads to a tremendous increase in susceptibility of infection to bacterial infections. And with anti-TGF-beta targeting medications, one could actually cause complete reversal of this increased susceptibility to infection in these models. So last, just to wrap up, and I think perhaps most important therapeutically, is to talk about what we can do about AMP deficiency. And this has been an area of interest way back to the time of Alexander Fleming. So he found the lysozyme, and not too much happened with that, so he kept plugging away, found that bread mold was useful. It had the same, his paper almost mirrors the lysozyme paper. He said these zones of lysis, and it got published and forgotten, that paper on penicillin. And then a couple of guys, Flory and Shane, who shared the Nobel Prize with him, were translational scientists, and they recognized that at the time of World War II, we needed something to help treat infections. There was a market for that. They, within, I think, 2 years from discovery of the raw molecule to mass distribution of the drug, they were able to show that penicillin was important, and we now know penicillin is important. So that's an AMP, but it's an AMP from a bread mold, not from mammals, but also important. In terms of the types of AMPs that we're thinking about that are peptides, there are examples of successful antibiotic use, and these are either synthetic versions of antimicrobials or naturally occurring that are nice now in the day, where microbes have shown they can beat any drug we throw at them, and rapid evolution of penicillin resistance has pushed our looking for these molecules that can be both antimicrobials, antibacterials, and antifungals. Another way to potentially treat AMP deficiency that we thought about early on, especially with atopic dermatitis, is can we somehow induce the natural expression of them from too low levels to something that was more appropriate for fighting infection. And one of the exciting observations made early on was that the human antimicrobial gene CAMP had a vitamin D responsive element in it. It was very responsive to the active form of vitamin D, and you can make a lot of it. So we were hopeful that vitamin D supplementation could in fact help patients with atopic dermatitis better regulate their AMP expression based on that. We did a very small trial, which was not successful, and what we also learned in animal models is that vitamin D active forms are very, very potent, very, very strictly regulated. So as you gave mice or you gave people more vitamin D, which is the inactive form, even though the inactive form might increase 100-fold, the body kept the active form just the same. So we really weren't changing the amount of 125 hydroxy vitamin D by giving more vitamin D supplementation. So I know that's still a very, maybe not so much as it was before. Maybe it is again, given the change in supplement thoughts. But it's hard to understand how oral vitamin D supplementation will have a tremendous effect based on the fact that the active form is so strictly regulated. Another drug that we observed that was very in this similar pathway, that vitamin D receptor, retinoid receptor pathway overlaps quite a bit, are retinoids. And indeed, in the case of acne, when you give isotretinoin, you see a big induction of antimicrobials in the skin. However, of course, retinoids, given their other effects on growth and differentiation and lipid synthesis, are certainly not going to be anything that I think we would ever try to use in patients with atopic dermatitis. The next was a surprise, and yeah, it works, and we all use it for treating our atopic dermatitis patients, and that is to inhibit the inhibitors. So early on, before dupilumab was actually approved, we started looking at, in collaboration with Regeneron, whether or not the patients who were receiving dupilumab in the phase 3 trials showed a change in their Staph aureus colonization. And Dr. Beck has also done a more recent study showing that it's phenomenal within only a few days of starting blocking of the Th2 system, the Staph aureus drops. Now, in animal models, it looks like that's because with the blocking of Th2, the antimicrobials increase. Humans that hasn't been confirmed yet, but we think that may be the case of why inhibition of Th2 is a mechanism for regulating bacterial colonization on the skin. And then lastly, what I just want to show you at the end is the other thought, and maybe it's sort of a simplistic approach, but if we have trouble changing the mammalian amps too much, and we know that there's a deficiency of the microbiome genes that are protecting against certain pathogens like Staph aureus, maybe we can just add more of the appropriate microbes that are producing the genes that make the amps. Just put it in a cream and rub it on the skin. So in this way, we've actually done now 3 independent trials in humans. The first was in that first publication where Dr. Nakasuchi observed the decrease in those, and this was a open trial where we just applied the cream containing one of the good bacteria that was missing in these patients, and within a day we saw a several log drop of the Staph aureus that colonized them. Then through funding by NIAMS with Dr. Hata, we did another trial where we looked for patient Staph aureus susceptibility and matched it to microbes that would kill that strain of Staph aureus. And in that trial, which was done in a placebo-controlled, double-blinded way in a single site, we saw that within only 7 days, there was over a log decrease in the colonization of Staph aureus, and there was a significant decrease by about 40% in the local EASY. And then a yet third independent double-blind placebo-controlled trial funded through the Atopic Dermatitis Research Network and NIAMS at 2 centers now at University of Colorado, at National Jewish Denver and at UCSD. We did a similar study, but now with a more realistic way to treat patients, found a candidate strain which is known as SHA9 that produces a very nice anti-staph set of molecules. And again, put it in a cream, rubbed it on their skin twice a day, and we saw that, again, across the population, the active cream caused over a 1 log decrease in Staph aureus colonization. And for the fraction of patients, which was slightly over 2/3 of them, that had Staph aureus that was susceptible to this strain of Staph hominis, they saw a significant decrease in their local EASI score by about 30%. And we were really surprised by this part. And, you know, these were basically phase 1 safety trials. We didn't think that shifting the microbial community would work so quickly after just a week. You know, this isn't a direct anti-inflammatory effect. This would be an indirect one, but yet, despite that, we saw a decrease in the local EASI at these sites. So that's about it. Just to summarize for you kind of the important points I hope you'll take from this talk. Antimicrobial peptides are an essential mechanism for host defense of every cell. Plants, insects, animals, they all use them. AMPs, particularly in mammals, amplify immune responses that can occur in the cases that I gave you through activation of G-coupled protein receptors and through scavenger receptor binding. The cell expression of AMPs is very diverse. It's a very complex and tissue-specific regulation. One of the processes that I alluded to is this dermal process where fibroblasts get triggered to turn into fat cells called reactive adipogenesis. And in that process, they make a boatload of AMPs like a neutrophil, and stress and other mechanisms inhibits that. And just, you know, I think we're only beginning to understand the relevance of this to human disease and how it influences what our barrier function properties are like and shapes the skin microbiome. So with that, I just want to thank a lot of people who have contributed to this work over the years and our current funding sources. Thank you very much.
I see a question burgeoning, unless it's a bathroom break. Okay.
It's really a great honor to hear your fabulous talk. And for your talk, I have 2 questions. It's very exciting to see the 3 small clinical trial about S. aureus killed. As in our clinical work, we also use the fusidic acid to kill the S. aureus. But from our clinical appearance, it does not work very well. And also, I noticed you mentioned the AMP deficiency. This seems very, for me, exciting. For the first question, I want to know which one do you think will be more promising for AD treatment, the AMP recovery or the S. aureus killing?
Yeah, that's the two great
questions.
More questions.
That's the first
okay.
question.
That's one question. Okay. It sounded like 2 questions to me, but I'll give you another one.
Yeah. For the second question, currently research shows that dupilumab treatment for atopic dermatitis can reduce the S. aureus earlier than the lesion improvement. It seems for me that inflammation is more important in AD pathogenesis. I just wondering for the AD treatment, especially for the recurrent AD, to inhibit AD recurrent in the future. Do you think the inflammation target is better, or the microbe target will be better for the AD treatment? Thank you. That's 2 questions.
Okay. What was the first question? No.
The first question,
why is fusidic acid not work that well? Got it. I'll answer the second question first, because I think that is kind of a little bit easier, and that is, it's both important. The itch signal can occur without inflammation, and we know that Staph aureus now produces proteases that trigger itch. Cytokines can trigger itch. Itch can result in scratch. Scratch can break the barrier and so forth. So it's clear that through a non-inflammatory signaling pathway, you can amplify aspects of the disease. The inflammation itself is also knocking out these beneficial bacteria, so it is negative. So when you ask what is more important, I think it's a cycle. I think addressing one aspect of this imbalance is not going to be as good as addressing both. And in the case of Th2 blockade, we see evidence that it is, in fact, addressing both. So to me, which is better is not relevant if it's a cycle. Regarding the fusidic acid efficacy in antibiotics, back 20 years ago when it was observed that Staph aureus colonization increased in atopic, there were a lot of different trials that looked at antibiotic, topical as well as oral interventions. And most of those did not work. And the ones that did not work rarely actually bothered to check if it decreased the Staph aureus. And if you check within the couple studies that did, observed somewhat surprisingly that even topical antimicrobials did not significantly reduce the Staph aureus colonization. So why an oral antibiotic or a topical, even though it might be active against the strain of Staph aureus, did not reduce the Staph aureus itself, has to possibly to do with the structure and the way those antimicrobials are working. So I think a lot of the failures of antimicrobial therapy, pharmaceutical type antimicrobial therapy in atopic dermatitis, had to do with a failure of actually being antimicrobial.
Okay, thank you.
Thank you so much.
Thanks so much for the wonderful talk. I always learn so much from you. My question is, for the antimicrobial peptides, if you purely, in conditions that are deficient in antimicrobial peptides, if you restored a level of AMPs, like with LL37 and atopic derm, how much does that recreate a healthy skin microbiome just by itself? Like how much weight does that component cause versus including Th2 blockade as well?
Yeah, so are you asking restoring it by adding the bacteria therapy or restoring it by trying to induce it with a hormone or something
Just
like that?
purely like, just the peptides themselves.
Oh, just add the peptides
Yes.
on it. We haven't done that experiment. I focused on antimicrobials because that's what we do. There's antimicrobial lipids. There's a lot of different components actually to that barrier. So adding just one is not going to make the overall balance of the microbial, moisture, all of those things. So it's unlikely, I think, that if there was still significant barrier breakdown, that even though you would kill pathogens with some of the targeted genes, that the rest of the community would be restored to exactly the same as it would be on normal skin.
Hello, thank you for the wonderful talk. My name is Wong Kin Lok from Hong Kong, and I just want to ask about alarmins. So alarmins in the pharmaceutical industry has been a good therapy, big interest in therapeutic targets for atopic dermatitis. I was just wondering about your opinions on alarmin inhibition, such as IL-33, TSLP inhibitions on the AMPs that you were discussing, and whether there are reports on AMP dropping or increasing in the atopic dermatitis patients, and what's your opinion on it?
Yeah, that's a good question. So a little history on that. The term alarmin was coined by Joop Oppenheim at the NIH, and he coined it to be applied to that slide that I showed you with LL37 activating the GPCRs. So actually, LL37 is one type of alarmin. It's a signal to mount an inflammatory response. And I'm not. It's probably been done, but I'm not aware of studies that specifically looked at some of the anti-alarmin effects on regulating these. TSLP induction and so forth is something that, interestingly, is induced by vitamin D. That's one of the reasons why we think MC903 works in animal models to make a Th2 skewed environment. So that may in part be early on making too much of the alarmin, and then later on skewing it Th2, so the IL-4, IL-13 pathways then start suppressing the expression. So timing becomes important when thinking about these therapeutically.
All right. Thank you so much.
Yeah. Thank you.
Thank you, Dr. Gallo. And speaking of alarmins, I, in my head, have an alarm going on right now because it's time for you to all go to break. Please come back in about 10 minutes. We'll start at 10 o'clock sharp, and we have really a host of equally exciting talks that's coming up on how our immune system turns off the alarm, as well as the different endotypes in our patients with AD, and how our therapies may have differential infection risk, followed by flash talks, lots on transcriptomics. So lots of exciting things. Remember, 9.5 minutes, so grab some coffee, go to the bathroom in any order, and we'll see you back here shortly at 10.
All right, so I think it's time to move on. Please take your seats because we are now starting the second part of our symposium. Again, thanks so much to all of you for coming, and it is my great pleasure to introduce Dr. Michael Howell. Dr. Howell was really instrumental in bringing two big realms of atopic dermatitis together. You know, we're always talking about barrier, we're always talking about the immune system. He really was very instrumental in better understanding how those two components work together by studying type 2 inflammation and the barrier. He is now in San Diego. He has decades of experience in atopic dermatitis research. He's the founder and president of Mountaineer Biosciences, co-founder of Galileo Biosystems, and a very important and trusted scientific advisor, and he has facilitated many drug developments that were then brought to market, and it is our great pleasure to hear him talk today.
All right. So it says 2 hours on here. Perfect. Just want to make sure everybody's paying attention. Thank you to the organizers for the opportunity to do this, to be able to come and give a little bit of a talk on research that's been focus, or at least a part of my focus for the last, I'm going to date myself incredibly here, but over 25 years. Very passionate about atopic dermatitis. One of the things that happens when you have kids is you realize that they ultimately develop some of the disorders that you had as a kid as well. And when my child actually developed eczema, it was something that was very near and dear to my heart. So excited to talk to you guys today. A couple of disclosures. None of these are relevant for the conversation today, but just in case they do come up, I'm a co-founder of a couple of different companies out there and have worked on different therapeutics that we're going to reference a little bit as well. So for those who are just coming back from the break, you're starting to think through what are we actually going to accomplish today. We've already had some amazing speakers that have talked a little bit about atopic dermatitis, the challenges within the barrier function, and what's actually going on within an atopic individual. I want to refresh your memory a little bit on some of the antimicrobial peptide classes that are out there, and I can already see people gravitating a little bit to the right. We'll come to that. But ultimately, there are several different classes that are here. You have the alpha defensins. There's roughly 4 of them. They're often involved in the neutrophils. They're usually about 29 to 35 amino acids, so they're relatively small for the most part. They do have differential activity, and that's part of what you're going to see across this entire spectrum, is that there's not just one class of antimicrobial peptides that's going to go through and play a role within your innate immune response. There are several different ones that come together. Ultimately, Dr. Gallo actually gave a really nice presentation. He and I collaborated on a lot of the work that we did for the cathalicidins, human cathalicidin as well. Dermcidin, I was actually relatively shocked that he didn't bring this up given the amount of work that his lab has actually done in this space. And then obviously there's more classes than just this, but when you look at the S100 proteins, this is a misunderstood class in my opinion. They do have antimicrobial activity, they're calcium sensing, and what we've started to see is that they do play a role within the antimicrobial activity as well. You'll notice on the right side of it, when in Rome or when in Chicago, you have to talk about one of the greatest defenses of all time. And we're talking about the 90s Bulls, 6 championships. Anybody who wants to negate that and thinks that somebody else is the GOAT, please see us afterwards. We can have that conversation. But ultimately, just want to talk about the first line of defense as we go through this, and really that's where the antimicrobials fit in. So what actually happens? You've seen a little bit here and there through the last couple of presentations. Ultimately, when you look at the atopic individuals, and part of the reason why we're susceptible or why they're susceptible to invading pathogens, whether it be bacteria or whether it be a virus of some sort, is largely due to, let's see if this works, love it, is the deficiency in a number of these different classes. So obviously, cathelicidin is very low in atopic dermatitis lesions, human beta defensin 2 as well, defensin 3, this is more of an inducible, the A7, which is elevated in psoriasis primarily, which is why it's called psoriasin, and you do see some level of elevation within atopic individuals comparatively back, but functionally it doesn't play a role. CCL20, for those who don't know, this is MIP3 alpha. It was shown to actually have antiviral activity and plays a role in recruiting Th17 cells to the site of inflammation. And then obviously dermcidin. So there are a number of different classes that we can actually review as we go through this process. It's not as simple as just saying, well, we were able to restore LL37. The human biological pathways are such that there's massive redundancies, complementarity that allows us to go through and fight off invading pathogens on a normal basis. So I remember when we first started talking about atopic dermatitis, and this is one of those stories of a long time ago in a galaxy far, far away, where when we would look and everything on this screen was dominated by IL-4 and IL-13. That was quite a while ago. We're now in the space where we're starting to see a number of different pathways that are playing a significant role within atopic dermatitis. And what is the relative contribution of each one of these pathways to the epidermal barrier, not only in just producing the antimicrobial peptides, but also in actual disruption by itself. And we've seen a number of these different pathways go forward. IL-18, which is a target of novel therapeutics, IL-25s, these are currently emerging in therapeutics. IL-1 beta, those have been tested for a while. We had a question earlier about TSLP and 33, we'll address that in a little bit as well. So we're starting to see the complexity and the heterogeneity within patients with atopic dermatitis, that this isn't just an IL-4 and IL-13 only driven disease. There's a number of different pathways. I just looked out, saw Nate Archer, so obviously the IL-36, characterized using a lot of his models as well. So ultimately, the reason why I put this up there is that there's no one-size-fits-all solution for most patients. What we have to recognize as we start to think through this entire paradigm is that we're now emerging into a place where we have to think about the differential subtypes, which is exactly what we're going to hear about in a little bit. So I don't want to steal the thunder. Again, a large number of these therapeutics on the left side of it, these have emerged, and we'll walk through those a little bit as well. So again, for those who haven't actually been involved in this space for quite some time, there's a lot of work that's gone in over the last 20 years, really characterizing the effects of a lot of these Th2 cytokines, and I'm going to lump them all in Th2 cytokines because it's part of the talk title. And so when you start to look at it, IL-4, IL-13, largely overlapping in terms of their signaling capacities, and ultimately what they're doing. They end up leading to a downregulation of the epidermal barrier, so flagrant lorocrine. And then as you start to look at the innate immune response, you'll see, and we'll walk you through a couple of these slides too, that there's a downregulation of LL37, human beta defensins, etc. We also characterize a lot of what's actually going on within these groups as well. The one that's a little bit unheard of for most people is thinking about the IL-31 pathway. And there's an overlap between IL-31 and IL-4 in terms of the signaling cascade. You get the JAK-STAT signaling downstream. And so there's some overlap there. But one of the main reasons for the actual role of IL-31 in the actual antimicrobials is the itch-scratch cycle. And so when you go through and you start to perturb the skin into various capacities, you're inducing what Dr. Gallo already referred to as these alarmins, and how they actually play a role within the biology of the system itself. And you'll start to see that there's alterations within the actual AMP production. There we go. So what does this all mean? How do you actually characterize what's going on? Can we actually outline some of the specific capabilities of what's happening with Th2-directed cytokines in this capacity? On the far left side of it, so very early on, Pek Ong, when he was a postdoc actually and an MD fellow at National Jewish, went through and demonstrated that specifically within atopic lesions that there was reduced levels of LL37 and HBD2, human beta defensin 2. We then further went on to demonstrate that using pro-inflammatory cytokines that could be found in most conditions for psoriasis, interferon, TNF-alpha, that you could actually induce the expression of human beta defensin 3, one of the complementary ones that was an inducible, and that you ultimately get an additive effect by combining them. Interestingly, in the presence of IL-4 and IL-13, you actually see that this pro-inflammatory driving aspect of it is inhibited. And so it raised a number of different questions. Chicken or the egg? Which comes first? Do you get the pro-inflammation? Because we know that within the epidermal tissue, if you actually have some perturbation, you're going to get TNF-alpha that's going to be released. Interferon and chronic atopic dermatitis is part of the elevations there as well. but IL-4 and IL-13 are pretty continuous throughout most of the lesion. So what actually happens? We're going to get there. In addition, we also looked at viruses. So beyond just looking at a cytokine stimulation and being able to show that the cytokine milieu was responsible for some of these effects, we demonstrated that if you use essentially a TLR3 driven, and we did experiments with poly-IC to confirm this. But if you actually do a exposure to vaccinia virus or other pox viruses that are out there, you'll start to see an elevation, specifically in the level of IL-37. This is part of the body's natural response to go through and fight off an infection. In the presence of IL-4 and IL-13, you actually see that the IL-37 is inhibited. Just as a caveat, so that we can explain why this is important, is that if you all remember about 20 years ago when there was an effort to go through and vaccinate many of the individuals in the military against smallpox prior to their deployment, there was actually an issue where through the scarification process, anybody who had atopic dermatitis was excluded from the actual immunization. And largely it was due to the fact that in the absence of a natural innate immune system, the actual pox virus, even though it's an attenuated form, would take over their body and end up leading to a condition called eczema vaccinatum that would hospitalize them. On the right side, so again, trying to really understand what's going on within this condition, you start to think through, all right, well, let's have some fun. Let's play with siRNAs. Let's play with knockout mice. All of the standard tools that are available at our discretion in most cases to start understanding what actually happens. Long story short, on the far bottom right, you'll see SI-STAT6. And we actually confirmed this using animals, so STAT6 knockouts, and even more so looking at some of the other information. But ultimately, we demonstrated, and you want to see the big difference between this bar and this bar. And what you'll see is that if you knock out STAT6, you're capable of going through and alleviating any of the IL-4 and IL-13 driven reductions in the antimicrobials. So, because we would be remiss if we didn't actually put up a really complicated figure so that everybody can take a picture of it just for fun. So as you start to look through this entire process, you've got the pro-inflammatory cytokines on the left side of this. You can see the signaling through the JAK-STAT pathways, ultimately leading to NF-kappa B, triggering the kind of production of these antimicrobial peptides. On the right side of it, you see IL-4 and IL-13. And the end-all be-all is that ultimately you get this activation of STAT6, which is really important for several companies developing STAT6 inhibitors now, and leading to a shutting off. So it actually blocks the NF-kappa B from being turned on and ultimately abrogates any sort of antimicrobial peptides from being turned on as well. And so we think about this in a really unique process. There are several different steps along the way. Obviously, you have the binding, the phosphorylation. This inhibits NF-kappa B. And then the inflammation that is actually reduced, and then the net result is that you have a reduction in these antimicrobial peptides. And I realize I went through that really, really quickly. And the reason why is because the part of the talk that we're going to go through really in a kind of a greater fashion is, what's the effect of the therapeutics that we have these days? And obviously, over the last decade, so realizing that Dupixent has been approved for about 10 years. So prior to that, we were largely using corticosteroids, cyclosporin, calcineurin inhibitors to try and treat patients who had a massive overinflammation. Now we obviously, Now obviously we have Dupixent, which blocks the IL-4 and IL-13. On the right side of it, you have Numluvio, which will target the itch associated with this, along with some of the Th2. There's a recognition that IL-13 is one of the dominant cytokines within the Th2 axis, and then trying to go after broad inflammation with JAK inhibitors, because we recognize that, again, one size doesn't fit all, and every single patient has a different profile. So again, why is this all important? As you start to think through the antimicrobials, can we actually reverse this condition? And it's an area, from my perspective, that we've thought about for quite some time, but we still don't understand everything of what we're capable of doing. So on the far left side, you'll actually see that this is human tissue. So we did biopsies from patients, 2mm biopsy punches, to be able to go through and understand from these individuals what's the general expression of these antimicrobials, and if you actually incubate it with an anti-IL-13, what actually happens. And we find that there's an augmentation of this actual antimicrobial peptide being released from the skin. So if we cultured those biopsies in the presence of an IL-13 neutralizing antibody, voila, we get IL-37 that's induced. Fun story in a minute. We then actually went through and started looking at, all right, well, we were doing a lot of work with vaccinia virus. Could we actually go through and understand what was happening in these individuals? Obviously, the US military at the time was trying to figure out how did they vaccinate everybody as well. And so this led to a lot of efforts, and this corresponds with what we see within Staph aureus as well, just so that we're all on the same page. But then in the absence of a neutralizing antibody, so again, this was lesional tissue from patients with atopic dermatitis. If you added in vaccinia virus, you get high levels of vaccinia viral replication, low levels of IL-37. Really interesting correlation. As soon as you add in neutralizing antibodies to IL-4 and IL-13, the vaccinia virus comes down, IL-37 goes up. Those are the same patients. So a really nice correlation. We actually took this information, believe it or not, and you'll notice the date on this. This is 10 years before Dupixent's approval. We took it to pharma and said, listen, within atopic patients, we're recognizing that neutralization of IL-4 and IL-13 is incredibly important at restoring the epidermal barrier, being able to go through and augment the antimicrobial peptides in these patients and preventing Staph aureus infection. We strongly suggest that you actually develop a monoclonal antibody that would actually block this pathway. For those who don't know, they all told us we were out of our mind and that nobody in their right mind would actually develop a monoclonal antibody for anybody with atopic dermatitis. That worked out well. So, why do I say this? Is because when you look at dupilumab, so for example, this is the prototypic IL-4, IL-13 blocker. When you actually look at Staph aureus, so again, moving out of the in vitro cultures, moving out of the ex vivo cultures, and into an actual clinical trial, these are the Staph aureus colonization kind of rates or levels in patients with atopic dermatitis that were enrolled in the trial and treated with dupilumab. And you'll see that by week 4, you start to get almost down to a plateau, essentially reducing the levels by about 70 to 80% within a matter of weeks. Same thing happened, so Dr. Beck, I don't see in here anymore, but Dr. Beck had actually shown this within tralokinumab as well. It's actually one of the features that we incorporated when we were developing tralokinumab at MedImmune, is that we added in staph colonization requirements and records, rather than just looking at a reduction in the infections, which is part of the typical standard protocols. And so here you also demonstrate over a 16-week period that you have a significant reduction in the Staph aureus colonization as well. So there's Th2. We understand that from a Th2 side of it, there are things that are involved. We can actually reverse the Th2-ness within an individual. Not everybody is actually responsive to an anti-Th2. So when you look at most of the data that's out there, targeting either IL-4 or IL-13 or targeting IL-13 directly works in about 40 to 50% of patients. And when I say works, I mean getting to an IgA of 0 or 1, so clear or almost clear. That means there's about 50% of the population that has a differential activity. And so what we've started to realize is that there's these other cytokines or alarmins or chemokines, depending on how you actually want to refer to them, that play a role within the epithelial barrier. Most notably, we've been following TSLP now for close to 30 years based on its initial identification. This is secreted upon mechanical disruption. There's an involvement within Th2 biology where it actually drives a lot of this. One of the biggest biomarkers that we observe within atopic dermatitis is CCL17 TARC. It's actually driven in large part by TSLP and some of the other cytokines. And so you get this priming that's there. IL-33, there's mixed data that comes through. So depending on the biologics that are out there, and we'll talk about them a little bit, that have gone through this space demonstrating that there's some differential activity. What's really unique, and you can see this in some of the publications, I don't have them highlighted here today, but you'll find that TSLP tends to be elevated in the lesion and IL-33 is elevated in the perilesional tissue, suggesting that one, they don't overlap, but two, they might be incredibly complementary to one another in driving this amplification or this immune process. The other one that's coming forward now, and it's really interesting to see how this entire field has progressed, is IL-25, or the other name for it is IL-17E. This was initially brought up, I would say 15 to 20 years ago when everybody was really trying to understand the IL-17 family, but nobody had any idea what this was actually doing. We actually abandoned it in most cases. There were companies who started to look at this and then walked away from it because they thought it was convoluted or too complex. We're now finding that companies are actually developing therapies targeting this. They're currently in clinical trials for atopic dermatitis, and largely the reason for it is because they think that there's a larger role in the actual Th17 axis. So can you, because of the whole role of IL-17 itself, amplify the Th17 axis, which will inherently actually drive the antimicrobial peptides? And so this is really interesting. That is really fuzzy. I apologize. Hopefully that'll be fixed in post. So I'll walk you through it anyway. On the left side of your screen, this is normal human epidermal keratinocytes. and they're just cultured over time. So if you actually culture keratinocytes in the presence of increasing concentrations of calcium, you'll induce terminal differentiation, you'll actually get activation of filaggrin, loricrin, and a lot of the antimicrobial peptides. Interestingly, if you do this in the presence of TSLP, you see this reduction. So indicating that as you go through and start to look at some of these epithelial alarmins, and I'm going to use that word, epithelial alarmins, they actually drive in large components what's actually happening within the epithelial tissue by itself. And so you start to see reductions within the, so this is S100A7, and this is human beta defensin 2. Simultaneously, we actually did some work at National Jewish and showed that if you increase the concentration, and I realize this is on a logarithmic scale, and so just take that in with a grain of salt. These are several fold higher in each case. And so when you start to look at it, if you just incubate the keratinocyte cultures with TSLP prior to going through and giving them vaccinia virus or even Staph aureus, you find that there's an amplification, specifically within the proliferation of the virus or in the proliferation of the actual bacteria as well. So this is not fully understood. There's not a lot of data that's out there showing that treatment with tezepelumab, for example, leads to a reduction in the actual Staph aureus yet, but this is something that we can hope for in the future studies as they move forward. For those who have not seen what a competitive landscape looks like in atopic dermatitis right now, it is incredibly crowded. But the way that I take this as somebody who, when I started working in AD a long time ago, there was virtually nothing. We had topical therapeutics that were calcineurin inhibitors. We had topical therapeutics of basically whatever we could pull together to try and treat these patients. I mean, you name it, things were tried. In some ways good, some ways bad. And as you start to go through this process, and honestly it's already out of date, is that as you start to look through, there's a large number of people that are developing or companies that are developing oral therapeutics. We are starting to see the emergence of degraders. There's a lot of things that are in the preclinical phase, which over the next year are going to enter into clinical trials. The topical space, we obviously have a number of approvals at this point in time, and we've seen a lot of these fail off as well. And then also from the injectable standpoint, almost everything that's approved at this point is a Th2 targeting therapeutic. Again, working at about 40 to 50%, which means that there's a lot of opportunity as we think about going forward. And you're going to start to see the emergence of these bispecifics or trispecifics that Pfizer, for example, just went through and did. This gives us hope of what's actually coming for these patients, because it's not as simple as the psoriasis story of, well, it's an IL-17 driven disorder, target either IL-17 or 23. Instead, we have a, well, what phenotype are you? We're going to learn. And so this is the idea, is that we are an incredibly complex disorder and that we're trying to understand how do we best treat these individuals. So as I close out this part of the talk and get ready for some questions, I want to give you a visual of the way that we think about what's happening within these patients. So in the upper left, you have a Th2 inflammation. And there's still a debate, is it the outside in, is it the inside out? Ultimately, we do know that there's a complication of human genetics along with exposures, environment. And so we know, ultimately, there's an elevation of Th2. How we get there? You end up with colonization by Staph aureus, and it could be different strains that are ultimately producing different toxins. That's becoming very important as well. These toxins ultimately open it up, so they create pores within the skin. You get allergen penetration, leading to a true perturbation, the inflammation. Hopefully you guys can see that redness back there. And you end up with this feeding forward mechanism, the IL-4 and IL-13 amplification. So again, that milieu that plays with suppressing the innate immune response, compromising the skin barrier. You end up with all kinds of barrier dysfunction that leads to further antimicrobial peptide downregulation, and then this cycle continues. And so this is what we see right now within the field of atopic dermatitis. We see different approaches at different stages of the disease of trying to figure out how do we treat these individuals. Is there truly a one-size-fits-all? No. And ultimately what we see is that there may be different times for different things. Maybe a biologic at the very beginning to be able to go through and hit the disease appropriately, reduce the inflammation, and then a maintenance therapy along the way to prevent flares from coming through, or being able to target specifically the bacterias that are actually colonizing and driving the inflammation. So my last slide is just to go through with some provocative thoughts. One is that I still believe, in my firm opinion, that AMPs, so antimicrobial peptides, are an actual biomarker. One of the things that we should look at, not only just looking at the epidermal barrier and being able to show the improvement within transepidermal water loss, but one of the things that we should look at also beyond flagrant is, do we cause the immune system to get back to a normal homeostasis? Can we fight off those infections that are actually driving the flares, that are driving the disease pathogenesis? One of the things that comes out of this is Staph aureus. So we've already seen in several different clinical trials that they've moved away from just saying, hey, we see an adverse event as a infection, a skin infection. We move from that to, do we actually cause an improvement or a reduction in staph colonization? And this can be presented as colony-forming units, it can be presented as RNA, and it can also be changed from the percentage of patients who test positive for Staph aureus versus negative. But ultimately, we're looking at this as a supporting of the improvement within the antimicrobials. Speed of restoration. How quickly do you see this? This is really important for differentiation. We've started to watch as different companies now are talking about itch, and how quickly can we actually address the pruritus within these patients? How do we actually go through and augment the immune system and capacity? So Dr. Beck previously within the publication I showed demonstrated that very quickly you can see a reduction in the staph colonization, which may play a role in the itch, which may play a role in the actual pathogenesis of the disease. Some of these things with JAK inhibitors, because you're hitting multiple pathways, might be good, but obviously carry a caveat behind it. The data gaps. There is, honestly, if you scour through the manuscripts that are out there, and if you're a bioinformaticist, I'd love to chat with you. That is not my gift. So when you start to look at things, understanding all the different transcriptomic data that's available and trying to see from the publicly available databases what's actually happening with each of these therapies, there's a scarcity of data. A lot of people have focused truly on the epidermal barrier without realizing that there's another component of the innate immune system that we're forgetting in some of these patients. And last but not least, I'm a data person. I like big data. I like to work with people who have big data. And I want to be able to understand as much as possible what's actually happening. So when you look at the disease states themselves, as we have novel therapies, an anti-IL-18 or a binding protein, or we have the IL-25s, or we have a STAT6 inhibitors, IRAK4 degraders, what's actually happening as we move beyond just a, hey, we're going to block generalized IL-4 and IL-13 to getting into a more comprehensive blockade of the inflammation within these patients. And with that, I'd like to thank people over the last 25 years that I've collaborated with, worked with, as we've built up a large amount of data within this space. And I'll take any questions if I have time.
While you're, oh, I see an audience. Okay, go ahead.
Hi, Mike. Great talk. I always have in mind those data on the clinical trials against IL-33, DSLP, works in AD, doesn't work in AD, work in asthma. It's not that clear. What do you think?
It's a great question. It is a question that has plagued industry for quite some time. Why does an IL-13, for example, neutralization work in atopic dermatitis but has failed miserably within severe asthma? Why does an anti-TSLP work in asthma but doesn't work as well within atopic dermatitis? And I think what we're learning through that entire process is the constant colonization. There's differences within the actual bacterial colonization, not only the viral repertoire as well, and what the relative contribution is to those in driving a flare, for example. The analogous way to think about it from an asthma versus AD is that the flare for an atopic patient is almost equivalent to an acute exacerbation for a patient with severe asthma. In some cases, your flare from an AD might put you in the hospital because you need to be treated appropriately. Same thing for asthma. And we see that there's a complexity in the way that those come out depending on the tissue. You don't have to run, Larry.
Well, one of the interesting things is we follow a biomarker that we think is going to be really, track with the disease, and then we have interventions that give differentiation. So just to make it clear, people don't know what I'm talking about. Like TARC is was considered to be a highly reliable biomarker. It's used as a tool in Japan to follow clinical disease. Our JAK inhibitors don't influence TARC at all. In fact, sometimes it goes up, while our IL-4, 13, and 13 blockers decrease TARC, yet there's a differentiation between clinical response with the drugs and how they may impact that particular biomarker. You pointed out there's a lot of data deficit in relationship to the antimicrobial peptides, but that whole differentiation of clinical response with an oral JAK, which is not doing a variety of things, may not decrease cutaneous infections in the same way. What are your thoughts on that sort of differentiation of that pathway versus our IL-4 or 13 blockade?
It's a great question. Believe it or not, when you actually treat with a JAK inhibitor, and obviously for those who don't know, is at Insight working on a lot of the JAK inhibitors, including Opzelura. When you go through and you give a JAK inhibitor, remarkably what happens is that the levels of cytokines go up. So IL-6 goes up, IL-1 goes up, and IL-8 goes up. And so the standard thought process is that you're actually going to reduce them. That's actually not the case. And so there's this counteracting hypothesis that you're still getting some level of activation of various pathways. And you have to think a little bit further down. This is why one of the challenging things when you're looking at some of those kinetic kinase inhibitors is looking specifically for phosphorylation of the actual downstream pathway to make sure you're hitting it correctly. Because ultimately what's happening is you're getting this augmentation. Same thing actually happens with some of the biologics. IL-33, if you want to know the biomarker of IL-33, 33 goes up. TSLP does the same thing. So that's where I think, to your point, we do have a massive data deficit. I don't think we fully understand the differentiation across the different therapeutic approaches and how they're affecting the biomarkers. A massive meta-analysis is needed. I would suggest that for anybody writing a grant. There is an interest in that. So as you start to think through it, I think hopefully it'll actually help us learn what are truly the biomarkers even beyond. Like periostin moves in the same way, it's IL-13 dependent. We can use that every once in a while, but there's differences. Great question.
Great. Thank you so much, Dr. Howell. That was terrific. Well, it's my pleasure to introduce onto the stage my co-chair for the symposium, Dr. Patrick Brenner. Dr. Brenner is the director of Cutaneous Lymphoma Clinic at Icahn School of Medicine in New York, and he is a clinician scientist with a strong interest in skin immunology who sees patients who are suffering from a number of different immune-mediated disorders, including atopic dermatitis, but also vitiligo, as well as, of course, cutaneous immune cell malignancies. And he is an NIH-funded investigator and also an avid oboist. I hope he doesn't mind that I share that. And we're so excited to have him to talk about a topic I think that's extremely relevant to what we think about every day with regards to AD, not only clinical care for patients, but also relevant to our current scientific endeavors in this area.
Thank you so much for this very kind introduction, and I'm very excited to talk about atopic dermatitis disease heterogeneity. One thing that we all wonder, everyone who treats atopic dermatitis patients will agree, there's no one size fits all. There's still a lot unknown, despite all the progress that has been made in treating those patients, so there's still a lot to do. These are my disclosures. So when we talk about atopic dermatitis heterogeneity, I think one of the biggest, aspects of it that we still do not understand is, so why do some children outgrow the disease while other children persist during the disease? You all know that atopic dermatitis typically starts early in life, and a good proportion of children lose their skin disease, can come back later. But up until today, we don't really know why some patients have persisting disease into for their whole lives, whereas others spontaneously outgrow their condition. Maybe it's the microbiome at birth or anything that those children encounter in their early life, but we still don't know. So traditionally, it's been talked about a lot today. There are, when we talk about atopic dermatitis pathophysiology, there's always two realms. There's the barrier and there's the immune dysfunction. So an immunity that is out of control that causes the skin symptoms that you see, which then impairs the barrier. And there are certain subsets of atopic dermatitis patients, filaggrin mutated patients, that have an epidermal barrier defect to begin with, and that leaky epidermal barrier likely facilitates inflammation. There are these inside-out, outside-in hypotheses, but most likely it's much more complex. And I think today we know that these two components of atopic dermatitis are tightly intertwined. So when we talk about AD phenotypes and endotypes, I think one important thing to talk about is like a genetic background and also ethnicities of patients. I already said there's a difference between children that outgrow the disease versus children that persist the disease into adulthood. We don't know really why this happens. When we looked at pediatric atopic dermatitis, there were more innate responses. There was more of a type 17 response in children. And when you look at various genetic backgrounds, various ethnicities, it is interesting to see that, for example, filaggrin mutations are primarily present in individuals with European descent, less so in individuals with African descent. And when you look at the immune, particularly the adaptive immune pathway, there is some nuance between those different populations. There's more of a Th17 or type 17 immune response in patients with atopic dermatitis from East Asia, whereas you get more of a type 22 response in patients from African descent. But what sort of unifies all those patients is there is a type 2 Th2 backbone that you find in all populations or have been described in all populations that we have been looking at. You already heard a lot about Staph aureus, Staph epidermidis, how it influences inflammation, and will be very interesting to see in the future whether those differences in children versus adults have some prognostic factors as to spontaneous resolution or persistence of disease, but I think we are not there yet. To sum all of this up, there are several attempts to map out atopic dermatitis to better understand atopic dermatitis heterogeneity. Again, there's a Th2 background and various contributions of other innate and adaptive responses, Th22, Th17, Th1. Also depends whether you have acute lesions, which seem to be less of Th1, Th22 strong, whereas once you get chronification of a lesion, there seems to be an intensification of all of these axes. Then, depending on the genetic background, filaggrin mutations, you have more or less contribution of these innate responses and barrier responses, and then, of course, the microbiome that seems to play a major role. So when you map out all of these aspects, you might think that, well, depending on what is up or down regulated, we're going to tailor our treatments. And this is a very, of course, very interesting aspect to think of, but I think we are not there yet. Many of the markers that are upregulated in certain subsets actually failed in clinical trials, such as, I don't know, IL-5 blockade doesn't really work, IL-17 blockade doesn't really work in atopic dermatitis. So I think there's still a lot to learn, and we must not forget only because markers are upregulated doesn't mean that those markers are truly the disease drivers. If we extrapolate from psoriasis, for example, there have been times, or for quite some time, psoriasis was characterized as a Th1 or type 1 disease, but blocking of that arm didn't do anything, actually made certain mouse models worse, and now we know it's type 17 inflammation. So there might be a yin-yang between those immune arms, and some of those immune axes might be upregulated because they counteract type 2 immunity, and when you take them out, you even get further exacerbation of the disease. So only because sometimes it's upregulated doesn't mean it has a disease-driving effect. It can also be counter-regulatory, of course. And this is, I think, why we are not quite there yet at precision medicine. Everyone's talking about it, but we are not really doing it. We are still doing trial and error, and we are still doing sort of standardized treatment approaches, mild, moderate, severe AD, and we introduce one medication, and once it doesn't work, we are switching to the next one. And we will see whether, with the advent of now more targeted treatments, IL-22, OX40, and so on, whether we will better understand whether there are certain subsets that respond better or worse. But still, I think we're still totally in the one-size-fits-all strategy, or one medication after the other. So when we talk about atopic dermatitis heterogeneity, one aspect is not only that AD is a heterogeneous disease, we also want to make sure that we are looking at the right patient, that we are making the proper diagnosis, because there are several conditions that can mimic atopic dermatitis, that can mimic eczema, such as cutaneous lymphomas, and when those patients are treated, they can respond quite differently to various medications. So there, I believe, are still conditions that are not very well characterized, that can be a mimicker of atopic dermatitis, and I think it's important to sort them out and to profile them, even if those are rare conditions. So we had an interest in patients that had adult onset erythroderma, and when you have a 70-year-old male that suddenly turns red from head to toe, you're of course always worried about Sézary syndrome or cutaneous lymphomas. But then you take one biopsy after the other, and nothing really shows up. There's no clonal rearrangement, there are no Sézary cells, there's nothing to corroborate it in the skin or in the blood. And there are certain patients that don't have atopic dermatitis, that don't have Sézary syndrome or erythrodermic mycosis fungoides, that don't have PRP, that don't have erythrodermic psoriasis, and so on, and you end up with the term chronic idiopathic erythroderma. And this was one population that we studied and were interested in, and we did some single-cell RNA sequencing studies with these patients and mapped out their T cells. So we believed those patients had a T-cell mediated condition because they responded to cyclosporin, but of course that's not a medication that you can give endlessly. So when we map out the T-cell cluster in the single-cell data set, one dot is one cell, we can not only assess the transcriptome of those cells, we can also map out the T-cell receptor, and we can check whether there's clonal expansion of certain T cells. And here we stained the polyclonal T cell infiltrate within healthy controls, atopic dermatitis, chronic idiopathic erythroderma, and erythrodermic cutaneous lymphomas, the polyclonal infiltrate in green and the top expanded clone in a color. And here you can see massive clonal expansion of a single malignant clone in cutaneous lymphomas. When you look at atopic dermatitis, we now know there's a bit of an oligoclonal expansion of T cells in chronic inflammatory skin diseases, including atopic dermatitis. But here we have a percentage of the top or most abundant clone mapped out, massive expansion lymphoma. Little bit of expansion here, that's not much different among conditions, but still there seems to be some specific clustering of those oligoclonal expanded T cells, and we took a closer look at it and found that in chronic idiopathic erythroderma, first of all, there's a different clustering from atopic dermatitis, but there was also a specific more of type 1 Th1 cytotoxic immune profile in those cells. So to make a long story short, no type 2 skewing, more of a type 1 skewing in those patients, and we believe that those are a separate entity and are still tough to treat. I'm a big fan of methotrexate for these patients with chronic idiopathic erythroderma because it seems to work quite well for them, but takes a long time to respond. And when you compare the skin and the blood of these patients, you find some low-level expansion both in the skin and in the blood of these erythroderma patients. And there was one marker that popped up, NKG2D. Those cells were abundant in the skin and in the blood. It's a killer molecule. Maybe that's some sort of misguided neoplastic response that happens in those patients, but that seems to be unique to those adult patients with erythroderma where we couldn't make a different diagnosis. So when we look at these various cells in those rare diseases, I think those are good model diseases to study certain subsets that we do not only encounter in those conditions, but also in atopic dermatitis. Everyone has been focused on type 2 or Th2 cells, of course. There are many other cell populations that are now being investigated in atopic dermatitis with particular biologic agents. Some had worked, some had not worked. Again, the T cell is for sure in the focus of therapeutic response. There's one cell population that gained some interest a while ago. Those are so-called innate lymphoid cells. which are cells that are part of the innate immune system, but they sort of mimic adaptive T cells. They are type 1, type 2, and type 3 innate lymphoid cells, and they produce interferon gamma, type 2 and type 17 cytokines. And those cells were thought to be important in atopic dermatitis, although they are present only at very, very small frequencies compared to Th2 T cells. So I'm not so sure they play that much of a role in atopic dermatitis because they're only at tiny amounts. The only disease where I encountered larger amounts of innate lymphoid cells was small plaque psoriasis. So now you're going to ask me, why are you boring you with this outdated term that no one, sort of some authors even say it's not appropriate to use anymore, because it's like all cutaneous lymphoma. You know, I had those patients in my clinic. They don't really respond to certain biologics. Some even think all of them have some sort of low-level smoldering lymphoma. So we took a closer look at them. The current notion is that a large plaque psoriasis is nothing but early stage mycosis fungoides, which I agree, but I think small plaque psoriasis is a separate entity that's completely different from atopic dermatitis or large plaque psoriasis. Because we were studying those patients, we were looking at clonalities. Clonality was quite abundant in large plaque psoriasis, not so much in small plaque psoriasis, and I think if you don't have clonality, if you don't have an expanded clone at a certain level, I think it's very difficult to call that condition a lymphoma. And I think single-cell RNA sequencing now allows us to look at the whole repertoire of T cells in the skin, and I think it's a very exact and a very sensitive and specific way to look at the T cell repertoire and quantify the T cell repertoire in the skin, I think much better than next generation sequencing of bulk material or rearrangement studies. But to make a long story short, we found some very consistent populations of an ILC1 population that expressed neuropeptide Y. We don't know why they express neuropeptide Y, and they don't fit any category. But if we corroborated those cells in immunohistochemistry, we found those cells to be present only in psoriasis and not in MF, healthy controls, or atopic dermatitis. And if you want to approximate them, they're most sort of similar to type 1 ILCs, but we don't really know what they do there. But I think if we talk about innate lymphoid cells, that's really the condition where I think we have some evidence that they might play a role as opposed to others. But what cells are sort of central in atopic dermatitis? You already heard a lot about antimicrobial defense today, lots of pathognomonic important cells in atopic dermatitis. We were interested when you treat atopic dermatitis with dupilumab and when the skin calms down, which cell actually remains there, which is the cell that mediates some immune memory in those patients. Because once you stop any treatment in atopic dermatitis, all of the disease will come back. And we found that a certain subset of type 2 cell or Th2 cells, so-called Th2A cells, are sort of one of the most prominent memory cells hibernating in the skin, even when you treat those patients with dupilumab, for example. And you heard a lot about alamines, so we shouldn't call them alamines, but nonetheless, those Th2A cells have receptors to IL-22, IL-33, and TSLP on their surface, and that's what really characterizes these cells. Those are memory cells that sit in your skin. You can't get rid of them, even with targeted treatments over 1 year of completely silenced skin. And those cells might be important mediators of disease recurrence, because you can, it's I think conceivable to think that when you take away any treatment, when you have those alamine receptor positive cells sitting in the skin, you just scratch your epidermis a little bit, you produce your alamines, and those cells might be able to wake up very fast and mediate a relapse. And these Th2A cells are really only in bona fide atopic dermatitis. We didn't find them in psoriasis. We didn't find them in mycosis fungoides or other conditions that we looked at. And I think those are central to the condition, and of course might be an attractive target. If you want to destroy those cells, get rid of those cells, you might even be able to induce long-term remission in atopic dermatitis patients. there's some circumstantial evidence that those cells are not just bystanders, but might indeed be important, because when you look at peanut sensitized patients, when you treat them with peanut specific, or when you do immunotherapy with them, and when patients lose their peanut allergy, those cells go down in the skin. So another piece of evidence, at least circumstantial evidence, that those cells might be important to atopic dermatitis. And one interesting aspect was, while those Th2A cells survive any treatment in your skin, when you look at individuals that have lost their atopic dermatitis, so we looked at young adults that had moderate to severe atopic dermatitis in their childhood but spontaneously cleared their condition, those Th2A cells are no longer found in their skin, in their adult skin. So there are, of course, two possibilities. Either their body got rid of Th2A cells, that's why they sort of are in spontaneous remission, or they never developed those cells in the first place. And that's, of course, also something that we are looking further into currently. So when we talk about atopic dermatitis heterogeneity, I think we should not only talk about, or we of course do not need only talk about, treatment responses. We also need to talk about response to treatment. And everyone who treats atopic dermatitis patients, that certain targeted treatments can have some paradoxical pro-inflammatory reactions. We see it in psoriasis, but we also see it in atopic dermatitis. You can get blepharitis, conjunctivitis with type 2 blockers, but you also can get this head-neck dermatitis in a few % of patients. That is not just flaring eczema or residual eczema, but truly a new rash, a novel or a rash that newly appears during type 2 blockade. And we took a closer look at those patients, and there's one interesting phenomenon in atopic dermatitis when you look at the T-cell clonal repertoire. When you do single-cell RNA sequencing, when you quantify your T-cell receptor repertoire and you look at clonal expansion, we saw, and others also saw, that you get some oligoclonal expansion within chronic inflammatory diseases, including atopic dermatitis. And when you take, for example, you can quantify the top 10 abundant clones in the skin, and you characterize them. And what we saw was that in patients with this head-neck dermatitis, you get a bit of an outgrow of certain oligoclones. It's not crazy high. It's by no means in the order of magnitude that you see in cutaneous lymphoma patients, but still there's some oligoclonal outgrowth, not that high, but very, very consistent, and also clustering very consistently in our data set, so I don't think that it's just stochastic or by chance. I think there seems to be some specific proliferation of those oligoclones in head-neck dermatitis patients more than in atopic dermatitis. And when you look at the cytokine production profile of those cells, they still produce tons of interleukin-13, so they have type 2 features, but they also start producing tons of interleukin-22 as well as the transcription factor AHR. So they seem to be sort of a mixed phenotype between type 2 and type 22 cells, and they produce this IL-22 cytokine at levels that we didn't see in other body parts or in sort of untreated atopic dermatitis. So that you could speculate that this head-neck dermatitis appears because you take away type 2 inflammation, not so much that you reduce the production of IL-13, but it, of course, cannot signal downstream anymore, and there seems to be disturbance in some sort of equilibrium that you otherwise have, and maybe type 2 inflammation is necessary to sort of counteract other arms of the immune system, at least in those patients. And we were able to corroborate our finding in immunohistochemistry, and those cells, IL-22 producing cells, were interestingly enough mostly of a CD8 phenotype and not so much of a CD4 phenotype. I know we're in an antimicrobial session, so you might ask, why am I talking about all of these things? So just to come back to microbes, this head-neck dermatitis has, we don't really know why it comes, might be those oligoclones. It could also be a response to malassezia, because when you treat those patients, there are some reports that antifungals can help in those patients, and if you want to speculate why you have those cells in your facial skin, it might well be that those are T cells that are there to mount an antimicrobial response in the face against malassezia, for example. And when you take away IL-4 and IL-13 signaling, maybe those cells start waking up and producing this rash. So there might be a crosstalk here, but of course that needs to be substantiated. With this slide, I want to thank my group, all our collaborators, and again, thanks so much for the opportunity to talk today, and thanks so much for coming. Thank you.
Great. This talk is open for questions. Patrick, really wonderful talk, and when you're thinking about these different phenotypes, I was wondering what you think about emerging field of sort of classifications of patients into different categories in terms of diagnostic innovations in that area, and how that could help us potentially clinically, either before initiating therapy or switching therapy. Do you feel like they have a role, or do those techniques need more validation, and so forth?
No, I think it's an enormously important aspect to look at. You could play devil's advocate and say, no, we just need the golden bullet. We just need one, cytokine that we target similar to psoriasis. And we were talking about stratified medicine in psoriasis, but everyone's just using IL-23 and IL-17 blockers. I think that's not the case for atopic dermatitis, because it's just such broader spectrum, and as long as we haven't found the golden bullet, the one cytokine that blocks everything, I think we don't have any evidence for that. And you just have a much broader variety. I think we will also need to decide whether we really call everything atopic dermatitis, or whether we might need to carve out separate diagnoses which might have a totally different pathophysiology, because we're still using terminology that is 300 years old. So there might be worth to revisit whether we can have some molecular mapping, and we're going to just subset that disease, and maybe if the pathophysiology is different in a certain subset, but the phenotype is the same, I think we just will need to come to an agreement whether we call it all eczema, or we're going to stratify, whether we split or lump the whole area.
Great, wonderful. And I see a question there.
Really wonderful talk. Vicky Fang, coming from J&J and UPenn. These Th2A memory cells, do they look like resident T cells at all? I know CD4 resident cells are less studied than CD8s. And if they're resident, should we be targeting them as sort of tissue resident memory?
Right, so those are, actually I didn't talk about all, so we found three populations. One is the DCLM positive DC population, then a CRTM CD8 positive population, as well as this Th2A. So those are CD4 positive T cells that have some unique marker expression, particularly those alamine receptors. If there is a way to destroy those cells, that might, of course, be a great approach. But getting rid of tissue-resident memory is very, very difficult. Those cells are designed by default to be very persistent and survive in the skin. The only thing, you're not even getting rid of them when you do allogeneic stem cell transplantation. So yes, if you would find a target to just destroy all these cells, maybe you push the reset button and get rid of your atopic dermatitis, or they might be refueled from somewhere else, but seem to be tissue resident memory T cells.
So they do look like tissue RMs, like CD69, maybe 103. They do express
yes,
those.
absolutely.
Interesting, thanks.
Okay, thanks for your great talk. And I have two questions. For the first question is about your sense immunology research. You noticed the Th2A, TC2 play a role in the dupilumab treatment, and even after treatment, they stay in the skin all the time. And for the natural immunology research, for the atopic dupilumab-associated head and neck erythema, you found the IL-22 cells placement. This cytokine plays an important role. I'm just wondering why the Th2A, TC2, that cells don't play a role for this kind of lesions since they stay in the skin all the time? That's the first question. For the second question, from your research, I realized the atopic dermatitis is so complex and heterogeneous, and I think in the future, precision treatment will be the main way for us to treat the AD. And according to your research, is the way we can use like the different biomarker or immune biomarkers or some combined markers for us so we can get the precision treatment in the future? These two questions.
Right, so as to the precision medicine, I think in clinical practice we're not there yet, but lots of people investing a lot of time and efforts into finding those biomarkers, and of course that's something that would be very desirable to test before and then treat it. As to your first question, I think with, when you talk about TH1, TH2, TH17, there's for sure much more plasticity. I think it's a very simplified and very rigid approach that might just not hold true. From ILCs, we know they can be very plastic. One can turn into the other. Now with single-cell RNA sequencing coming up, when you look at individual cells, so those so-called TH2 cells, they all produce IL-22 and they also all produce IL-26 to a certain level. IL-26 previously more attributed to type 17 inflammation. So there seems to be much more plasticity going on over time, I think. It's not that every cell can turn into each cell, but there seems to be some plasticity there, and also maybe because some targeted treatments work in one patient or not the other, maybe there are certain escape mechanisms where one cell, okay, my IL-13 can't signal anymore, let's turn up IL-22. I think what we know from neoplastic diseases might just be totally the same in chronic inflammatory diseases, that at least those 20-30% of patients that are difficult to treat, maybe they just then turn, or most likely it's just going to turn up other sort of pro-inflammatory mediators sort of as a compensatory mechanism because certain immune circuits are interrupted and other ones are taking over.
Okay, thanks.
Really great talk, Patrick. I just have a question about the T-cell profiles for the dupilumab-associated head and neck dermatitis and whether or not there were any similarities. I don't know if you've looked at cases of the lymphoid reactions due to dupilumab and also dupilumab-associated CTCL, whether there's some similarities between those and whether, for example, if you can comment, I know AHR and IL-13 are also important in CTCL and related kind of reactions.
So I think like this lymphoid hyperplasias or lymphoid reactions, I think are completely different animals. So we also had a few of these patients. They suddenly have a CD4 to massively increased CD4 to CD8 ratio. All those cells are CD30 positive, but you don't really find a clone and you don't really have loss of antigen. So I think that's a rare but completely different situation that I think has nothing to do with that kind of head neck dermatitis, because in those lymphoid reactions really a CD4 process, and those clones in the head neck dermatitis were all CD8. Yeah.
Great. Thank you very much, Dr. Brenner.
And now it's my great pleasure to introduce Dr. Armstrong, who is a professor and chair at UCLA. I'm sure she doesn't need a lot of introduction. She's a giant in the field of eczema and psoriasis, chronic inflammatory skin diseases, and really has a big passion not only for chronic inflammatory skin diseases and clinical trials. She's also a well-respected and well-loved educator. She has published numerous papers, has participated in many clinical trials, and we're very excited to hear from you today.
Thank you. Wonderful. Thank you, Patrick, for that kind introduction. And today I'm going to talk about mostly regarding clinical evidence. We just had a great morning thinking about the translational work in this area. And you must be wondering, how does it bore out in terms of our epidemiologic evidence as well as in clinical trials? There's a slight theme through my talk. How many of you have seen the Avengers movies? Okay, so about 80% of you, great. I'm sorry for the 20% of you who have not seen them, but we'll get started. So we're gonna look at the infection risk that's associated with our currently approved atopic dermatitis therapies, and hopefully I think some of the themes will relate to some of the translational work that you've learned earlier. Here are my disclosures. And I'm going to first talk about the infection risk that may be inherent in patients with atopic dermatitis, regardless of therapy. And then second, I'm going to dive deep into some of the different therapeutic classes and what are some of the clinical trial results in terms of the infection risk, as well as some of the real-world data. Okay, so for those of you who have seen the Avengers, you probably remember the first Avengers movie, which was back in 2012. Oh, no conflict of interest with Avengers series. And you probably remember it was in New York City where Dr. Brenner is. And this is the first time where you see some mayhem and destruction from the movie. And I think of this much like the disrupted AD barrier that we see with regards to impaired barrier dysfunction and as well as microbial assault. And when we're thinking about this and thinking about our epidemiologic data with regards to infection risk among AD patients, we are thinking about a few different aspects here. One is that we know that regardless of therapy, that we have about 50% increased risk in our AD patients in terms of herpes simplex virus infection risk. And if we look at serious infection risk, and in clinical trials, we typically define this as patients who have either had hospitalization due to their infection or they have had received IV antibiotics, is that we see about anywhere between a quarter to about 40% increase in terms of the serious infection risk. And then finally, when we look at the more severe risk among our AD patients with regards to infection, we also see increased rates of opportunistic infections. And this is important, and you may be asking, what are some of the opportunistic infections that our AD patients may experience? And they can include things such as Cryptococcus and also candidiasis, for example, Aspergillus, and histoplasmosis, and so forth. So they are at increased risk for these different types of infections. And if I can have our nice AV staff there to put my timer on, that would be great. You can subtract a few minutes for the times that I've used. So let's take a look at hospitalization rates. So on this graph, you see two lines. You see the top line, which represents patients, adult patients with atopic dermatitis, and these are, in the bottom line, regards to patients without AD. And we're looking at all comers, adults with AD, and you're looking at hospitalization due to serious infections. So what you see is that this is large epidemiologic data, specifically looking over time at the different rates over the years. And what you see is that there's a big difference between patients with AD who are hospitalized for their serious infections and then compared to patients without AD. Now, when you're looking further at this data, one of the things you may be wondering is, yes, of course, they may be hospitalized for things such as cellulitis or herpetic infections due to skin, but are there any extracutaneous serious infections that they may be at risk for beyond the skin? And one of the things that was noted is that, yes, in other organ systems, they also have increased rates of serious infections requiring hospitalization. Now, most of this is due to respiratory tract infections. And when we're looking at this, especially lower respiratory tract infections have a higher likelihood of AD patients being hospitalized for those serious lower respiratory tract infections. There are also signals for GI infections as well as urinary tract infections with regards to our AD patients at independently increased risk for those infections. And then the strongest evidence, so this is mostly from the Danish cohort, where they follow everyone nationally, and it's a national system, so regardless of insurance, they're able to track them over a long period of time, and therefore able to find those infection risks. So it doesn't matter which hospital you go to, in this Danish cohort, they're able to track this. So we thought, wow, this is really important. We've been talking about cutaneous infections this morning, but now we're seeing in patients with AD in terms of extracutaneous infections that they're at increased risk for, and particularly these respiratory infections. So when we're thinking about infections in AD at the population level, we're recognizing that it's not just skin infections, but also especially some of the respiratory infections that they're seeing. And could this be the shared type 2 inflammation that is impairing the mucosal membrane that can in part explain why we see these increased rates of respiratory infections in our patients with atopic dermatitis? And so we want to be especially mindful in our patients with allergic comorbidities, so patients with asthma, for example, and as well because they may have altered mucosal injury, and these altered mucosal injury not only manifests in skin, but also in terms of these respiratory infections. Okay, now back to Avengers. But do not worry, we have multiple different therapies that are being both used as well as under investigation, as you see from Dr. Howell's talk earlier. If I think about the different Avenger characters, probably Hulk is a good representation of the JAK inhibitors. But if you don't like the Avengers analogy, let's go back to my former governor here back in the 80s when he was jacked. So let's get jacked. Okay, so in the United States, we have primarily upadacitinib and abrocitinib that's being FDA approved for a number of years now to treat patients with moderate to severe atopic dermatitis from ages 12 and above. But worldwide, we also have baricitinib that's approved in many other countries for AD. And when you look at these JAK inhibitors for AD, they have different JAK isoforms and different selectivity, and there are differences in terms of binding affinity, association constants, and also their relative affinity for the different members of the JAK family. So when we're thinking about these JAK inhibitors, one of the key discussion points is the infection risk associated with these JAK inhibitors. But before I talk about that, let's just think about general risks and the general annual risks before talking about infection risks. So this is based on epidemiologic data. So apparently, you have the likelihood of being struck by lightning of 0.01% if you look at the general population. You can die at home, also similar risk at 0.01%. Interestingly, I just want to point out your death from a motor vehicle accident is very similar. So whether you stay at home or go outside, it's about similar. So your rates of hospitalization from sports-related injury is about 10 times higher, 0.1%. That's why I don't do any sports right now. I'm just kidding. But as you can see, there are just a general baseline risks just as being human. And I tend to sort of explain this to my patients because as we know, our patients' ability to tolerate risk or estimate risk can be quite variable depending on who you're speaking with. So let's take a look at this upadacitinib example based on their Measure Up 1 and 2 clinical trial. So when we are looking at upadacitinib rates, and then this specifically talking about serious infection rates, what we see is that the 15 milligram serious infection rates is about 22 events per 1,000 patient years. So it's about 2.2 per 100 patient years. That's for the 15 milligram. If they go up to 30 milligram, what you see is that it's about 36 events per 1,000 patient years. So that's really important. So we learn, but to set this into context, if you look at the general moderate to severe AD population, regardless of therapy, the serious infection rates is about 11 to 13 IR per 1,000 patient years. So it's about 1.2, 1.3. So what we learn is that in this case, upadacitinib, the rates of serious infections are slightly increased compared to the general population. But probably the more also as equally important point is that if you do increase, if you use a higher dose of upadacitinib, your relative risk is increased as well. And here is a summary of the different JAK inhibitors with regards to their serious infection rates and herpes infection rates, which we'll dive into a little bit later on. But here are some of the raw numbers from the clinical trials. So let's take a look at the serious infection rates in context for our JAK inhibitors. A few things we noted is that we just reviewed the rates for upadacitinib here, dose-dependent. For abrocitinib, for the clinical trials, it's relatively similar, but some of the real-world studies have shown some dose dependence there as well, probably not surprising, around 2.3. So pretty similar, I would say, compared to the lower dose of upadacitinib. For baricitinib, at the doses that are approved in other parts of the world, not in the US, for AD population, it's a little bit less than 2.5%. So, and then some of the latest systematic review have shown perhaps this risk of serious infection was not statistically significantly elevated, but if you look at some of the placebo control periods where we have a placebo arm, you do see slightly increased risk of serious infections, but the absolute rates are still low. So I still counsel our patients on JAK inhibitors regarding that particular risk. Okay, so your superhero, ChatGPT did not allow me to use one of the actual superhero due to copyright reasons, apparently. But let's say your superhero asks, do JAK inhibitors confer a higher risk of herpes virus infection but this case, herpes simplex virus infection in your AD patients? Okay, how many of you think that our JAK inhibitors likely confer a bit higher risk for herpes simplex? So I'm not talking about herpes zoster simplex. Okay, I see some hands going up. So let's take a look at the data. So the magnitude of this herpes simplex virus risk is really dose dependent. So if you look at the clinical trials where we have the placebo group, and if you look at upadacitinib, for example, you have 2% for the placebo group, and then 4% for the 15 milligram, and 8% for the 30 milligram. So the risk when you're looking at the placebo control period are increased. And the real world data, if you look at the population data from the real world, what you see is that it's about 2% for abrocitinib, and then 6% for upadacitinib and baricitinib. So when you're looking at reasons for discontinuation, herpes simplex virus is actually one of the infections, one of the most common reasons for why patients discontinue from JAK inhibitors. And then when you look at both clinical trial data and some of the real world data, we do see an increase in that risk. I know we talk about zoster all the time, which I'll address a little bit later, but something to make note here. So what are some of the practical approaches for patients with or without a history of herpes simplex virus? So for those without a recurrent HSV history, routine prophylaxis currently is generally not supported. For those with recurrent HSV, oral or genital, or prior bouts of eczema herpeticum, you may consider valacyclovir with regards to daily suppressive therapy. Now, for patients with multiple bouts of eczema herpeticum in the past, you may want to consider the suppressive valacyclovir therapy in those patient populations. Now, this is especially true if you're using a higher dose of your JAK inhibitor, if you're using 30 milligrams of UPA or 200 milligrams of abrocitinib. Okay, so you're probably asking, well, what about zoster infection? We talk about herpes zoster infection all the time with regards to the risk in patients with JAK inhibitors. So let's take a look at, this is a 6-year upadacitinib data. If we look at the right-hand side, this is a herpes zoster rates. The green bars represent those who are on 15 milligrams, and the blue bars represent those that are on 30 milligrams, and we're looking at across the different years. So 2 things we learn from that. Overall, zoster rates with cumulative use, they don't seem to increase over time, so they stay relatively stable. However, if you are using 30 milligrams, as you can see, the infection risk is higher than if you're using 15 milligrams. And what about differences in ages, right? So this is looking at baricitinib and upadacitinib, looking at rates of herpes zoster based on their age group. And these are patients, who, so let's take a look at the different age categories. I'm a little bit upset at the demarcation of the age categories in this. As I get older, somehow I feel like, not to reveal my age publicly, but somehow the last bin starts at age 45. I thought it started at age 65. Anyhow, so what we see here is that as you get older, not surprisingly, your risk your risk of getting herpes zoster on these JAK inhibitors increase. So definitely make sure that we're going to talk about the zoster vaccine later on, but just recognize your older patients are a significantly higher risk. Okay, so let's talk about practical things in zoster vaccination. So it's recommended that for your adult population and 19 years and older, that they should definitely get zoster vaccination before starting the JAK inhibitor. Ideally before starting when feasible. However, oftentimes that may not be feasible. So they can start, for example, their first dose, because as you recall, it's 2 doses. So they can get their first dose before they start. You can start them on the JAK inhibitor. The second dose here is typically at month 2 through 6, anytime during that after they start the JAK inhibitor. I tend to do the second dose a little bit later on. There is no recommendation currently for routine hold of the JAK inhibitor. So that's a recommendation that's kind of wavered over the years, but the latest is that for the zoster vaccine, as it's a non-live vaccine, that you don't need to hold your JAK inhibitor with these doses. So you can go ahead, give one before you start, and then between month 2 to 6, then you can give the second shot while the patient is on the JAK inhibitor. Okay. Now you have a teen, right? You want to start potentially one of the JAK inhibitors. So what about zoster vaccine in those 18 and below? Well, let's think about this because this is a real clinically relevant question. And so what should be done? So first of all, before we even look at this population, it's important to know that in that population, they get oftentimes the varicella vaccine. And that varicella vaccine is a live vaccine, and that's contraindicated typically during JAK inhibitor therapy. Now, if you have to give it during the JAK inhibitor therapy, you do need to hold your JAK inhibitor 1 week before and 4 weeks after giving this live attenuated vaccine. So the most important part in your adolescent patients is to make sure that they've had their varicella vaccine series. And if they haven't, then you want to make sure that they get it before you start the JAK inhibitor. And so that's the main message. And you may want to verify they have the VZV immunity for adolescents who may not have, because you all have patients who don't have documented varicella vaccine. They say, well, I think I got it, but I don't know where my records are. So you may want to verify VZV immunity for those patients. For example, you are not quite sure if they got and they think they may have gotten it. All right, so we're going to switch gear in my remaining few minutes here to talk about IL-4 and IL-13 inhibitors. And let's go to dupilumab. I really like Dr. Howell's presentation earlier that looked at the Staph aureus counts and then the decrease with the dupilumab therapy. And here we're looking at the adjudicated skin infection. So this is looking at overall skin infection rates, including bacterial infections, including viral infections. And I'll walk you through this. You'll see 3 sets of bars for different ages. So as you go across the screen, you go from younger children all the way to adults. And you'll see overall kind of 3 sets of bars. The gray bar represent placebo group, and the blue bar represent Dupy Q2 week, which is our approved dosing, and then the purple bar represent Q weekly dosing, because in their studies they looked at that as well. So you'll notice something. You'll notice that the gray bars are a lot higher than the blue bars or the purple bars. So the overall skin infections, excluding the when they look at it, this particular one, excluding the herpetic infections, are much higher in terms of our placebo group. So I think that correlates really well with what we're looking at earlier. So it could be, number 1 is restoration of the barrier function, and therefore you get likely lower bacterial infection, but also perhaps some other reason beyond that, as we've been exploring with regards to IL-4, IL-13 inhibition, and the regulation of the Staph aureus immune defense system. So let's go on later to look at some of the herpes. So you're saying, what about herpetic viruses? And we'll dive into that story a little bit deeper in later slides. It's a little bit nuanced. So when we look at this, specifically herpetic infections, and this is looking at herpes simplex as well as herpes zoster, what we see is that it's a little bit variable. Your gray bars around the same height as your blue and purple bars. There's a little bit of variability there, but overall not a huge difference there. This is one of the more recent network meta-analysis looking at dupilumab in the global cohort compared to our older therapies such as methotrexate or cyclosporin. So you see the rates of the herpetic infections as well as systemic infections to the left of 1. So basically it's telling us dupilumab therapy in comparison to methotrexate or cyclosporin have lower rates of overall herpetic infections as well as overall infections. And then here we're looking at serious infection rates, looking at drug therapy, the specific medication, and also looking at some of the other medications with regards to lebrikizumab, tralokinumab, and nemolizumab. And I'll show you some of the highlights with regards to the serious infections. So overall, what we saw is that with dupilumab, based on the clinical trial, serious infections that can land your AD patients in the hospital or have them get outpatient IV therapies, Dupi is overall associated with a significant reduction in these serious infections. Lebrikizumab showed overall very similar to the placebo, and then tralokinumab also had numerically lower serious infections on tralo, and Nemo overall pretty similar to placebo as well. So probably this is consistent with your prior perception of these biologics. What about herpes simplex, eczema herpeticum, and herpes zoster rates? So those are a little bit more nuanced, and I will take you to some of the highlights that we learned in this area. So first of all, when you look at dupilumab and when you're looking at eczema herpeticum and herpes zoster, what they consider sort of clinically more relevant, important herpes infections, they did see a decrease overall relatively compared to the general population. But what they also saw is a slight numeric increase in the oral herpes in some of the studies. So for the oral herpes infections, the data is a little bit mixed. But overall, when we're looking at eczema herpeticum or zoster infections, there's no increase in the signal. Rather, actually, some of the studies support a decrease in those. For lebrikizumab, overall herpes rates were lower than, numerically lower than the placebo, as well in the long-term exposure-adjusted analysis. In its placebo control period, they did see numerically a few cases of herpes zoster that were seen in the lebri group that wasn't seen in the placebo group. But overall, when you're looking at the long-term data, there weren't an increase in terms of overall herpes infection. Tralokinumab showed lower rates of eczema herpeticum in the clinical studies, and for nemolizumab in the placebo-control period, they saw several cases zoster in the treated patients versus none in the placebo arm. But the long-term data for the overall infection as well as overall herpes infection has also been reassuring for nemolizumab as well. Okay, so you're probably wondering, April, you mentioned earlier about the increased rates of a lower respiratory tract infection just natively in our patients, regardless of therapy. What do our biologics show? So this is newer data looking at the various studies of our looking at both our biologics as well as some of our JAK inhibitors. Overall, when you're looking at, and this is mainly weighted towards, because of the way the study is done, mainly weighted towards the biologics, overall you don't see any increase with regards to upper respiratory tract infections in this particular analysis. And when you're looking at the other analyses, taking into account both upper and lower respiratory infections, you also don't see that with our biologics. I'm going to end with a topical therapy here because you're probably wondering, you didn't mention anything about topical therapies. And I will mention that to date, when you're looking at some of the RCTs of the topical steroids up to 6 weeks of time, overall you don't see an increase in skin infection during that short period of time. Our non-steroidal topicals, you also do not see with regards to increase in skin infections. This is an older network meta-analysis that had been done comparing topical steroids versus our topical calcineurin inhibitor, and they did not see a difference in terms of skin infection rates between those two groups. I'm going to end with one of our neurotherapies that's emerging, which is OX40 inhibitor. And but before that, just to note that as we know, Kaposi sarcoma is a HHV-8-induced endothelial tumor, and we have case reports of patients who have these inherited human OX40 deficiency who manifest with KS in childhood. And also we know that there are certain populations who may be at increased risk for having HHV-8. So one of our emerging therapies targeting OX40, you may have heard in the press, have reported events of Kaposi sarcoma. To date, they've been restricted or confined to patient populations who may be at high risk for contracting HHV-8, such as men who have sex with men. And then, and if you are following them over time, what you see is that resolution of the cutaneous lesions when they are discontinued from the therapy. So I just want to cover that part since that's a relevant topic here. So as you walk through Central Park, and hopefully your superhero characters have now starting to restore the balance in the world and restore in the skin. By the way, you may actually encounter characters like that at Central Park in real life. But I think we are hopefully having more and more therapeutic options that will help us combat not only infections, but also the different disease phenotypes that Dr. Brenner has mentioned. And I want to just thank my team. They're on the flight right now, arriving. They will be dressed in the, I'm just kidding, they will not be dressed. But I especially want to thank my wonderful fellow, Kimberly Smart, who has been working in this area with me. So I want to thank you all very much.
Thanks so much for this inspiring talk. Are there questions from the audience?
I only take your question if it's 3 parts.
Okay.
I'm just kidding. I'm kidding.
I only have one question for you. Thanks for your great talk. And as Dr. Patrick showed that the atopic dermatitis has many different immune phenotypes. But I just wondering for different AD phenotype, whether the JAK inhibitor treatment will induce different kinds of infection. Whether there are some different, and how we should balance the treatment choice between biologics and JAK inhibitors so we can avoid the different infection.
I think that's a wonderful question. To date, there hasn't been any rigorous studies that looks into differential characterization of these infections based on the different phenotypes of the patients that are treated with JAK inhibitors. Most of the studies have been mainly looking at their baseline demographics, thinking about their infection risk and so forth. So I think that's a wonderful kind of future study translational question that's quite relevant. So thank you. Excellent, great.
Just one question.
Yes.
So how are you handling like patients that have an infection, have a fever, don't have a fever, that are on a JAK inhibitor? What are you advising your patients to pause it, treat through? Because we know that JAK inhibitors have a positive effect on COVID-related inflammatory complications. So it's more complex than just yes or no.
Yes, absolutely. I think if I have a patient on when I have a patient on JAK inhibitor who develop a fever and active infection, I usually have them stop the JAK inhibitor and have them evaluated either by me or at urgent care because I want to make sure that I have a good idea of what is driving their infection. So overall, I do tend to hold their JAK inhibitor for a few days. But I do hear you about our COVID data with regards to the positive effect of JAK inhibitors on the cytokine storm that some of the patients may experience. Yeah.
Another question?
Yes.
That's a quick curiosity question. So what happens after you remove the anti-OX40 in the KS patient? Does it go away?
Yeah, so to date, they're still following those patients, but overall, what they see is that their KS lesions do resolve. Yeah. Okay, great. We're going to go on to the next part of our talk, our programming, and if I can have Eleanor and Katie start to come onto the podium, that would be great. We are going into our flash talks, and I just want to say this is a very, very competitive process in that we got many wonderful abstracts that we reviewed through our, that were submitted to the SID, and the next 3 talks that you'll hear were competitively selected from all the abstracts that have been reviewed. So Eleanor, yes, that's Eleanor, everyone. So Eleanor is a rising third-year medical student at Northwestern University Fine School of Medicine here, and her current research interests include cutaneous lymphoma as well as some of the visceral cancers, cutaneous metastasis. So thank you for being here. And then we have Katie next to her. So Katie is our rising 4th-year medical student at Texas A&M who is completing a research year at Northwestern University here in Chicago as well. So we're very happy to have them here, and they're going to talk to us about integrated epidermal microbial and spatial transcriptomic profiling. Come on up.
Hi, everyone. Thank you so much for the introduction. My name's Katie.
My name's Ellie.
And today we're excited to present our work titled Integrated Epidermal Microbial and Spatial Transcriptomic Profiling Reveals Staph aureus Associated Keratinocyte Programs in Erythrodermic Skin Disease. Here we go. Okay. One of the previous talks discussed erythroderma, but just to briefly review, it's an inflammatory skin condition characterized by diffuse erythema and scaling of the skin. Over 80% of the body surface area. It can arise from multiple etiologies, including cutaneous T-cell lymphoma, atopic dermatitis, psoriasis, drug rash, and autoimmune disease. And these can all clinically appear very similar, but the management approaches are very different among these different subtypes. Oh, there we go. Here are some photos of the diffuse erythema and scaling that we see, as well as the hyperkeratinization that we can also see with erythroderma. And as you can see, these patients present, like I said, very similarly, but the underlying etiology of their disease here are quite distinct, and so that motivated our study to look into how to differentiate the different types of erythroderma. So we analyzed 17 patients with erythroderma of varying subtypes, as well as 4 healthy controls, and then skin swabs underwent full-length 16S RNA sequencing to define the microbial composition, and then we took epidermis from lesional punch biopsies, and those were analyzed using nanostring geomix DSP for the keratinocyte profiling. And differentially expressed genes were those with an LFC greater than 0.58 and an FDR-adjusted p-value less than 0.1. So we'll first start with our microbiome data. One of the patterns we observed was a low microbial diversity state where 1 to 2 species tended to dominate these samples. And so we wanted to understand how Staph aureus abundance related to the epidermal biology of our samples. And what we saw was with samples that had high Staph aureus, there was upregulation of barrier and metabolic dysregulation genes, KRT14 and CKB respectively. And then we saw downregulation of apoptosis regulators, protein synthesis, and innate signaling. Just to note, KRT14 is found on undifferentiated or immature keratinocytes, and Staph aureus there's prior research that shows Staph aureus tends to disrupt the keratinocyte differentiation process. Thus, it would make sense why in our population, or in a high Staph aureus population, we would expect this upregulation of KRT14. We also wanted to look at Staph hominis abundance, and we saw a very different pattern here. We saw transcriptional programs that were associated with overall structural normalization, mitochondrial homeostasis, as well as immune quiescence. And then to note, we saw the downregulation of KRT14 in the high Staph hominis abundance. And then just to note, FLOT2 helps with membrane structure and differentiation, and then MUL1 is downregulated, and that is a mitochondrial stress response gene, and so that would be downregulated in these samples. And so we then wanted to look at the high microbial diversity states to see how these differ. And overall, we saw, first to note Shannon diversity, it looks at both the richness and evenness of the samples. So richness is the amount of species, the different types of species, and then the evenness is how well distributed the species are. And so here we saw a multitude of genes that were upregulated, mostly in regards to apoptosis regulation, TNF signaling, and keratinocyte differentiation, as well as vesicle trafficking. And then to note, we saw a number of genes downregulated here. TSLP is relevant in regards to atopic dermatitis. It plays a role in the vicious atopic Th2 signaling. And so in these higher diversity states, we saw downregulation of TSLP. And then we also looked at observed diversity, which just takes into account the richness, and we saw upregulations of genes related to the proliferative turnover, signaling and cytoskeleton organization, as well as immune surveillance. And we saw decreased regulation of genes in regards to cytoskeletal stress, inflammatory dysregulation, cellular stress, and autophagy, as well as metabolic stress. So overall, in these high diverse states, we saw, or high diversity states, we saw membrane stability. And so we wanted to look at the relative abundance across all the samples. And so in this plot, it's a little overwhelming at first, but to break it down, you see relative abundance at a genus level. We have healthy controls on the left, and you see a number of different colors showing the diverse microbiome that we would expect in healthy skin. And then if you look over towards the right of the plot where atopic dermatitis and CTCL are, you see this teal color really dominating the plot, and that represents Staphylococcus. And you overall just see a fewer number of colors in our erythrodermic skin subtypes. So we wanted to look more into the Staphylococcus since that tends to dominate the erythrodermic skin samples. And here we have a similar plot, but looking at a species level, a Staphylococcus species level. And we see on the left, the healthy controls have a relatively low amount of staph in general. And of the staph species they have, they tend to be commensal bacteria like Staph epidermidis and Staph hominis. But then if you look over at atopic dermatitis and CTCL, you see another teal color, but this time it's representing Staph aureus. Especially in the atopic dermatitis samples, you see the Staph aureus tends to dominate the microbiome there.
And I'm going to be going over our, spatial transcriptomic data. I just want a quick note for tech. Our timer dropped like 2 minutes, just so you know. But if we could maybe get those back, I don't know if we will. But this is our spatial transcriptomics, aka our geomics data. First, we looked at CTCL versus other erythroderma, and as you can see here, we have upregulation in CTCL-based erythroderma of genes related to CTCL and associated markers like CXCR4, BCL2L2, Siglec16. We also see upregulation of keratins like across the board, KRT16, 6A, 14, 1. And then I want to highlight specifically upregulation of YPI1, which is a master autophagy regulator linked to CXCR4, and FTH1, which is related to iron sequestration and is experimentally validated to confer ferroptosis resistance in CTCL. Meanwhile, downregulated in CTCL erythroderma, but upregulated in other erythrodermas by comparison, we see YUND and FOSL2, which are related to atopic derm, as well as HLA-F and HLA-DR-B1, related to immune surveillance and innate immunity, and IgHG3 related to humoral immunity. Notably, TSLP and GATA3 are not significant here at all, and that's highlighted just because we believe this is related to the fact that maybe all erythroderma might be a little more Th2 skewed. We also took a look at atopic dermatitis versus other erythrodermas. And here, there's not really much that's significant. We think this might just be because potentially the classical atopic dermatitis genes are not very upregulated across or different across all the erythroderma subtypes. But notably, in other erythrodermas compared to AD, we see elevated DOC3, which is a suppressor of toll-like receptor signaling, and S100A9, which has been extensively discussed this morning as an alarmin and defensin. So when we took a look at integrating our data, we did a reactome pathway analysis, which demonstrated that for CTCL keratinocytes, we see strong enrichment of innate immunity and barrier dysfunction transcriptional programs with weaker apoptotic and oncogenic signaling. And then in atopic dermatitis, we saw enrichment of apoptosis and transport-related signaling. In psoriasis, we saw enriched barrier dysregulation and TP53 signaling. And then for atopic derm versus psoriasis, we saw enriched apoptotic and innate immune pathways compared to psoriasis. So kind of putting all this together, keratinocyte marker correlations, markers correlate with microbiome state in terms of dysbiosis and eubiosis. You can see in Staph aureus on the left, there's pretty elevated markers that we've talked about previously, like FTH1, YPI1, CXCR4, keratins, but there's also a distinct profile for microbiome eubiosis, where all of these markers are then downregulated in Staph hominis, high Shannon diversity, and high observed diversity. Additionally, looking at keratinocyte marker expression across erythroderma diagnoses, there's a pretty distinct profile for each diagnosis you can see in this heat map, where CTCL has elevated CXCR4 and other associated markers, elevated keratins, while drug rash only has elevated exo-C6b, which is a vesicle trafficking protein, and psoriasis, on the other hand, has elevated serpin B3, and atopic dermatitis, which are two in the middle, have elevated keratins and YUND. Limitations of this work include small sample size. Unfortunately, we only had 21 patients, and only 4 of those were healthy controls, only 1 autoimmune disease patient. As a result, we had to expand our FDR-adjusted p-value threshold to 0.1. Additionally, geomics can sometimes capture immune infiltrate instead of what we're trying to look at, and have contamination. And lastly, we did only look at the keratinocytes, not just the dermal stroma. So in terms of future directions, we really hope this work can be utilized to create some sort of gene biomarker panel. And this is a proposed draft of what might be helpful for future research, because for each disease category you see here, there's a unique and easy to understand profile with selected markers that we thought were the most significant and useful for discriminating between diseases. I'm just going to let you all read this for a second. For our acknowledgements, we'd first and foremost like to thank Dr. Zhou, who is our research mentor, as well as Dr. Guittart, one of our other research mentors, and our co-authors, as well as our collaborators, our clinical team, and our patients. And this is our bibliography. We went techie. We have a QR code.
Is there one burning question? If not, then thank you so much for your presentation. Next is Dr. Tobin Dickerson, who is co-founder and CSO of Mindera Health, a biotechnology company in San Diego. So if we want to do all this profiling, all this stratified medicine, we also, of course, need sampling methods because we're not going to be able to biopsy every patient multiple times. So I think minimal or non-invasive sampling is really key for future directions, and I think we're going to hear more about it. Thank you.
Thank you very much. Thank you to the organizers for inviting me to speak. We've heard a lot about biomarkers so far and ways that we need to get access to biomarkers. And I'm going to spend a fair amount of my time talking about our platform and how we actually get access to these biomarkers and how we use that. In this case, the lens is going to be differentiating psoriasis from atopic dermatitis, but really being able to look at how you get molecular data in a non-invasive way. These are my disclosures. I am the founder of the company, which probably is the most relevant one of all of this. So why atopic dermatitis versus psoriasis? Quite frankly, when it first was presented to me, I wasn't sure there was a real need for this assay. And then I have a colleague who is also a recovering academic like myself here in the room, who reminded me that there are some real problems with this. And in particular, it comes up in clinical trials and in drug development, where you have between 10 and even up to 20% in some trials of patients who are misclassified up front. And really what the issue there for me, and what I worry more about is, yes, the costs go up, but it's the confounding data that really causes the real problems here. So there is a need here to be able to actually really identify which patient you're really looking at. So how do we do it? So we've developed a platform called the Dermal Biomarker Patch. You see sort of a zoom in there on the, let's see if this works. Oh, it does. There, that sort of zoom in and set there. So it's about the size of a penny. The center of it is 5 by 5 millimeters, where you see those small projections. Those projections are not hollow. You're not pulling out fluid. You're not pulling out tissue. They're actually chemically modified on their surface. So they're chemically modified to bind specifically to biomarkers of interest. So what we do is we bind to the transcriptome. So we bind to RNA, specifically mRNA. And the way it's applied is with this sort of single-use, easy applicator that you see here. It's as simple as unlock and press down to apply a patch. What does it do? It collects molecules. So it collects the molecules, it preserves them, and when I say preserve, we've seen that we have RNA stability at room temperature for weeks when bound to these patches. So really flying in the face of much dogma. Surrogate for biopsy, great overlap with biopsy. And then we run this through our laboratory that was mentioned in San Diego. So we run a CAP-CLIA laboratory out of San Diego where we can process samples. The one you see here has this logo. That is actually, if you're familiar with our company, is a commercialized product that we use for predicting the best drug for psoriasis patients. But this platform can be applicable to anything in translational science. It's something that Dr. Howell and I have been working on for the past few months on launching this under subtilitis. So what does this patch do when it's in the skin? It's no pain, no bleeding, no scarring, no keloid formation. And really you're binding, like I said, to the transcriptome, getting about 7,000 biomarkers per patch. And then on the back end, we're using RNA-seq and machine learning to really understand questions of the data. So the way I like to think about this is almost like a hardware-software approach. We have some hardware that's unique. It's easy to use, and it can be used in a non-invasive manner. You're not destroying the skin that you're sampling. And then on the back end, we use RNA-seq and machine learning methods and data science to ask questions of that data. And we can ask any question. We can ask diagnostic questions. We ask predictive questions. And in this case, we're asking a differentiation question, almost like a stratification question. Excellent work, quantity and quality of RNA comes out of these samples. The reason I go through all of this, I should have said this up front, we've been trying to bring this to more studies than just psoriasis and atopic dermatitis. So it's important, I think, to understand the quality of these samples and how it can be used in any study. And I encourage anyone who wants to talk later, either Mike or myself, are more than willing to talk about all kinds of studies that can be used with these samples. Excellent concentration, easy to go straight into sequencing, able to make libraries, able to reproduce samples or replicate samples. And like I said, stability is excellent with this, where you get very long-term stability. These were done at 4 degrees. You can do this even warmer. Like I said, we've done this at room temperature and seen no degradation of samples. It seems to be a function of directly of the patch itself. It's having RNA bound directly to the patch. Okay, and then why I'm here today. So using some of our internal data sets, we were looking at the difference between atopic dermatitis and psoriasis. We have 2 data sets that are internal to us. We actually have a larger, but this is sort of a culled data set for psoriasis in particular, where you see we have 21 atopic dermatitis patients and 50 psoriasis patients in this set with relatively similar characteristics, leaning more towards more moderate or severe cases in the psoriasis case, to be able to really look at differences between those 2 indications. And this is a bit of an eye chart, but I think one thing I like about it is that even though you can see, so samples are up here, this is patients, these are genes, and the segregation, it doesn't take a whole lot of effort to be able to see the segregation between the 2 groups. This is not using any sort of classifier build yet. This is not doing anything but just clustering these. And you see, so psoriasis are the orange, atopic dermatitis in the green. They segregate very, very nicely. The genes we see there are as expected, and I'll talk about that in a second, about what kind of genes we see and what we're doing with this data. But really, it shows just a stark difference between the 2 indications from this patch, validating what is known from biopsy, validating what is known in the literature. And you can take this and actually feed this into some other platforms. So we use the CyberMed iSort platform that's been recently launched. It's a platform that lets you put in transcriptomic data, and it tries to extract out cell types, understand more than just pathway analysis, but what kind of cell types you're seeing. So what we did is compared some normal, so we have healthy controls, psoriasis and atopic dermatitis you see on the right graph. It segregates out and tells you what kinds of cells you're seeing there, whether it's neutrophils, eosinophils, that sort of thing. And what you see is exactly as expected, and sort of belaboring this a little bit, this is a lot of text, but the psoriasis biomarkers are dominated by the IL-17 axis like you would expect. The genes we see align very much with what you've known from the literature, whereas the AD biomarkers show a Th2 skewed axis. This to us is really validating. It's showing what's already been known out there. Dr. Howell can speak to this as well, that there's been data that he worked on at a previous company similarly showing that you can see this kind of difference. The challenge has always been, how do you get access to the molecules in a way that's easy? It's not easy to get access to molecules when you have to use biopsy or other methods that have been out there in the past, like tape stripping. So next steps, where are we going with this? This is newer data that is not using that data set I just showed. This is what we like to do with this kind of data set. So this is a Dupy-treated data set where we're able to start to see differences between treatment and non-treatment with Dupy. We like to be able to predict, what we do in psoriasis, we predict what drug response is going to look like prior to exposure. What's needed for this, and I think it's supported by some of the other speakers earlier, atopic dermatitis is incredibly heterogeneous. We see the same heterogeneity, and it's going to need a much, much larger data set. But what we want to do is continue working on this, because even with this limited data set, this is just an N of 10, you can very easily see that there's starting to get separation between these 2 groups. And based on my timer, I have 10 seconds left. So I think I can run inside Congress. I can reserve my time for the next speaker.
Well, thank you so much for this talk. So when you separate eczema and psoriasis, when you look at those ones, I believe psoriasis and eczema and vice versa, did you go back to the clinic and challenge the diagnosis, or were there certain atypical features of those patients where, you know, it might have been a different diagnosis in retrospect?
Yeah, so the reason that's a culled set from our psoriasis data set, which is probably closer to 500 patients, those were culled specifically for confirmed diagnoses that were very classic, where we did challenge at least those. On the atopic dermatitis front, not completely challenged, but I would say we've done it as close as we can now, knowing that there was no atypical features in them. It doesn't mean, that's where I would like to do more, quite frankly. We did a small study years ago and saw essentially reverse phenotypes over time.
And at the same time, you might, you know, I'm sure the atypical features might be most interesting, because you might be able to pick up those and, you know, stratify for potential different treatment approaches.
No, I think, like I said, we've seen already in some patients where you see complete reversal of trends, and they're definitely with atypical features, that's why. So that just becomes it's a game of ends. Now, I need more samples, more partners to work with.
Are there any other questions? If not, thank you so much.
Thank you.
Thanks. And our final talk comes from Young-Woon Lee from Rockefeller University. She's a clinical scholar there. That's actually a wonderful program. I myself was a clinical scholar at Rockefeller, so if you're interested in translational research or you want to become an MD scientist, that's the place to go. And she's going to talk about compartment-specific transcriptional profiling defines two distinct subgroups in atopic dermatitis psoriasis overlap phenotypes.
Thank you for the kind introduction and the opportunity to share our study. So I will be presenting on spatial transcriptomics in patients with atopic dermatitis and psoriasis overlapping phenotypes. So to set the stage, atopic dermatitis and psoriasis have classically, traditionally been viewed as immunologically distinct diseases. So atopic dermatitis is driven by Th2 and psoriasis is characterized by Th17 axis, which is the basis for the current targeted biologic therapies. However, in clinical practice, this dichotomy is increasingly blurred. We often see patients with psoriasis with eczematous features or atopic dermatitis with psoriasiform morphology. And also we also experience biologic-induced phenotype switching. And all these observations suggest that atopic dermatitis and psoriasis may exist along a broader inflammatory spectrum than previously appreciated. So despite this growing recognition of overlaps, there are still problems. Treatment decisions are often difficult in these patients because targeted biologics are highly axis-specific, and we do not have full understanding underlying these unresolved biology. So our aim was to define compartment-specific immune profiles in these clinically ambiguous cases and translate that into something diagnostically and therapeutically useful. So for our study, we included total 41 lesional skin biopsies, including 19 overlapping phenotype cases, and we conducted a spatial transcriptomics using GeoMx profiler in 14 patients, and we validated the findings with immunohistochemistry in an expanded cohort of 35 biopsies, and 8 of them were also included in the transcription cohort. So overlap cases were histologically and clinically shared the features of atopic dermatitis and psoriasis. For example, this patient showed a eczematous plaque resembling nummular eczema, but some other lesions resembling small plaque psoriasis. So on the PC analysis, you can see that the reds are atopic dermatitis and the purple ones are psoriasis, and they separate really clearly. However, overlap ROIs kind of spread around. So some are overlapping with atopic dermatitis and some are leaning more towards to psoriasis. Using differentially expressed genes between classic atopic dermatitis and classic psoriasis, hierarchical clustering shows that in the middle, the overlaps shows differing expression patterns. So this suggests that the AD and psoriasis overlap segregates into 2 molecular subtypes. So we call overlap 1, which are aligning more towards the atopic dermatitis, and overlap 2 towards psoriasis-like. Next, we moved on to examine the biologic pathways. Overlap group 1 was enriched with cornified envelope and glycosphingolipid metabolism in the epidermal compartment, and neurexin, neuroligin, and sensory perception pathway in dermis. On contrary, overlap group 2, which was psoriasis-like, was enriched with pro-inflammatory pathways, including IL-1, IL-15, 17, and interferon and neutrophil degranulation, resembling psoriasis. So gene scoring further reinforced these findings, overlap as a whole. So on the left figure shows that it sits intermediate, like it's in between atopic dermatitis and psoriasis. When the subgroup in this subgroup analysis on the right, you can see that overlap 1 is sits aligning with the atopic dermatitis with Th2 high signatures, while overlap group 2 shows Th17 and interferon gamma high, resembling along with the psoriasis. So we next move on to find biomarkers that can stratify these two molecular subgroups. On the right figure, these are the list of the transcripts that we thought it was important in their underlying biology, differentiating the AD-like and psoriasis-like overlap groups. So atopic dermatitis and overlap group 1 was increased with the CCL13, which is Th2 cell chemokine, chemotrectant, and its receptor CCR4, and also a cluster of neuropeptide and sensory signaling molecules, including neuropeptide B. In contrast, psoriasis and O2 high markers included MAVs, with a gene called MAVs. It is involved in the type 1 interferon induction, and also C1YF43, which is involved in the keratinocyte differentiation. So based on these findings, we moved on for immunohistochemical validation, and so we stained these MAVs and C1YF43 in an extended cohort with 35 biopsies, and then we try to classify these overlaps into AD-like and psoriasis-like based on staining intensities relevant to the classic AD and psoriasis. So importantly, I don't know whether you can see it clearly, but there is a red dots in this bar graph. These are the patients included in the transcriptomic cohort. So they were, so the subgroups, the patients in the overlap group 1 and 2 in the transcriptomic experiment were also concordantly well divided into O1 and O2 by the immunohistochemistry. And we also further validated this finding with additional staining with IL-36 gamma, established psoriasis marker, and showed that the overlap group 2 and the psoriasis showed higher staining intensity compared to the overlap 1. So we believe that this gives us a translational implication. Overlap group 1 may benefit more to Th2 targeted therapies, while overlap group 2 may respond better to Th17 targeted therapies, or these overlap groups may benefit from broader acting agents, including JAK inhibitors or TYK2 inhibitors, or emerging bispecific antibodies targeting both Th2 and the Th17 spectrums. So in conclusion, clinically ambiguous overlaps are not a single uniform entity, and we believe that the biomarker may, in the future, guide the treatment options in these overlapping phenotypes. So I'm done with the time, but I would like to appreciate Dr. Jung-Eun Kim and Sun-Pyo Jin for leading the study, and Dr. Jae-Won Kim and Dr. Kruger for their invaluable support. Thank you for your attention.
A quick question. In the world of translational research, those of us who on the more clinical arm always like to push back on, or at least ask questions about the clinical description and the phenotyping or endotyping. So the question is, were these natural patients who had combination features of psoriasis and atopic dermatitis, or any of these patients were atopic dermatitis patients who became psoriasiform with other therapies?
Oh, thank you for the great question. So for our study, we excluded biologic treated patients. So they were all biologic naive patients. Yeah.
Any other burning question? If not, thank you so much.
Thank you.
All right, I think we had a great time this morning. What a great start to the SID meeting, and I want to thank all of you for being here and for staying the entire time. I want to thank all the speakers who did a wonderful job and really appreciate all that. I also want to thank on the back also Jennifer Westland and as well as Josephine Fazio, and of course, Alan Stiles. Thank you for, they're really the people who really help organize this and putting this together. Thanks, Larry, for leading the organization, and I want to thank Patrick for being co-chair. And with that, please scan this. It takes about a minute for the evaluation. We take our evaluations very, very seriously, and it helps us to improve our meetings for the future. So I want to thank you all for your attendance here. I hope you learned a lot, and we will see you around the conference. Thank you.
Thank you so much. It was a great joy having you all here. Please look at the Eczema Council website. There are several symposia coming up. And yeah, let's keep in touch. Thank you so much.